Reproduction is the defining line between the living and the non-living. It's the one biological function not required for your survival, only for the continuation of your species. A single human begins as two cells, sperm and egg, merging in a moment of biological precision. What follows is perhaps the most intricate process in all of nature, the creation of a new individual. Over the next few hours, we'll explore this remarkable journey from specialized cells to independent life, examining the systems, hormones, and mechanisms that make human reproduction possible. Let's begin. Reproduction stands alone among biological processes. Unlike respiration, circulation, digestion, or neural function, reproduction is not required for an individual organism to survive. You can live a full, healthy life without ever reproducing, yet reproduction is absolutely essential at the species level. Without it, there would be no next generation, no continuation of genetic lineages, no future. This paradox, essential yet individually optional, makes reproduction unique in biology. The evolutionary imperative to reproduce has shaped nearly every aspect of life on Earth. Organisms have developed extraordinary strategies to ensure their genes pass to the next generation. Some species reproduce asexually, creating identical copies of themselves through processes like budding or binary fission. This approach is efficient and reliable, requiring no mate, no courtship, no elaborate rituals. A single bacterium can become millions in hours. A hydra can sprout a clone of itself from its side. Simple, straightforward, effective. But sexual reproduction, the kind humans engage in, takes a dramatically different approach. It requires two parents, two sets of genetic material, and the fusion of specialized reproductive cells called gametes. This system is far more complex, far more energetically expensive, and far less efficient than asexual reproduction. So why did it evolve? The answer lies in genetic diversity. Sexual reproduction shuffles genetic material with each generation, creating offspring that are genetically unique, different from either parent, and different from each sibling. This variation provides raw material for natural selection. In a changing environment, populations with greater genetic diversity stand a better chance of survival. Some individuals will possess traits that allow them to thrive under new conditions. Pathogens that evolve to attack one genetic profile may be thwarted by another. Sexual reproduction is essentially a long-term survival strategy, trading efficiency for adaptability. The human reproductive system represents millions of years of evolutionary refinement. It's not designed for maximum efficiency. Conception is actually rather difficult, requiring precise timing and a cascade of successful events. But the system produces genetically diverse offspring, equipped with immune systems primed by both parents, bodies adapted through millennia of selection, and the potential to thrive in varied conditions. At its core, human reproduction requires two types of gametes: sperm cells produced by males and egg cells, properly called oocytes or ova, produced by females. These are not ordinary body cells. While most human cells contain 46 chromosomes, 23 pairs, gametes contain only 23 chromosomes, half the normal number. They are haploid cells, created through a specialized form of cell division called meiosis. When sperm and egg unite during fertilization, they restore the full complement of 46 chromosomes, combining genetic information from both parents into a unique new individual. The production of these specialized cells requires specialized organs. In humans, these are the gonads, testes in males, ovaries in females, but producing gametes is only the beginning. Those cells must be matured, stored, transported, and delivered to precisely the right location at precisely the right time. They must be supported with nutrients and protective fluids. They must be guided by hormones operating on schedules ranging from minutes to months. And in females, the system must be prepared not just to create a gamete, but to nurture a developing embryo for 9 months, then deliver and nourish an infant. This requires not just gonads, but an entire reproductive system. Ducts and tubes for transport, glands that secrete supportive fluids, organs that house and nourish developing life, hormone producing structures that coordinate the timing of everything, and nervous system connections that respond to stimuli and coordinate muscular actions. The male reproductive system is organized around continuous production and delivery. From puberty onward, males produce millions of sperm cells daily, storing them until they're needed, ready to deliver them on relatively short notice. The system is externally accessible, with primary structures located partially outside the body cavity for temperature regulation. The female reproductive system follows a different design philosophy. Females are born with all the egg cells they will ever have, a finite supply that decreases throughout life. Rather than continuous production, the female system operates in cycles, preparing one egg each month for potential fertilization. The structures are entirely internal, protected within the pelvic cavity. And the system must be prepared not just for gamete production and fertilization, but for the possibility of pregnancy, a commitment lasting the better part of a year. These fundamental differences, continuous versus cyclical, external versus internal, quantity versus preparation, reflect different reproductive strategies. But both systems share common goals: produce viable gametes, facilitate their union, and provide the best possible chance for successful creation of a new individual. The creation of life requires more than anatomy. It requires chemistry in the form of hormones, signaling molecules that travel through the bloodstream, coordinating events across the entire body. The hypothalamus in the brain produces releasing hormones. The pituitary gland responds with stimulating hormones. The gonads produce sex hormones that affect reproductive organs, influence behavior, and feed back to the brain, creating loops of regulation that maintain balance. Reproduction also requires behavior. Humans must feel motivated to engage in sexual activity. They must be capable of arousal. The male must achieve erection, the female adequate lubrication. Both must coordinate their actions. While the biological mechanisms of reproduction can be studied clinically, the behavioral dimension reminds us that reproduction is not merely mechanical. It's influenced by emotion, attraction, psychology, and social context, factors that extend far beyond anatomy and physiology. The timeline of reproduction spans decades. It begins before birth, when the gonads form during fetal development. It pauses during childhood. It activates at puberty, when hormones surge and secondary sexual characteristics develop. It operates throughout reproductive years, with males capable of fertility from puberty into old age, and females cycling monthly from menarche around age 12 until menopause around age 50. And the act of reproduction itself spans from the moment of intercourse through fertilization, implantation, 9 months of gestation, labor, delivery, and into the months of nursing that follow birth. At every stage, things can go wrong. Gametes may be damaged or malformed. Hormones may be imbalanced. Structural abnormalities may prevent transport or fertilization. Genetic errors may cause developmental failure. The implantation site may be abnormal. pregnancy may end prematurely. Each successful birth represents hundreds of processes proceeding correctly, a testament to the robustness of the reproductive system despite its complexity. Understanding human reproduction means understanding this complexity. It means tracing the development of specialized cells from their origins, following their journeys through elaborate anatomical pathways, recognizing the hormones that orchestrate timing, appreciating the moments where sperm meets egg, where embryo meets uterine wall, where fetus becomes infant, and acknowledging that this process, refined over millions of years of evolution, represents one of biology's supreme achievements, the ability of life to create more life, generation after generation, each individual unique, each carrying forward the genetic legacy of countless ancestors. Over the coming hours, we'll explore this system in detail. We'll begin with male anatomy and the production of sperm, then turn to female anatomy and the creation of eggs. We'll examine the hormones that regulate both systems, the act of intercourse, and the moment of fertilization. We'll follow early development as a single cell becomes an embryo, then a fetus. We'll see how the placenta forms to nourish that growing life and how the maternal body adapts to support pregnancy. Finally, we'll witness birth, the dramatic transition from life inside the womb to independent existence. This is not a simple story, but it is perhaps the most fundamental story in biology. Let's begin by examining the male reproductive system, where millions of specialized cells are produced each day in preparation for their singular purpose. The male reproductive system is organized with remarkable efficiency around a single goal: produce sperm cells and deliver them to a location where they might encounter an egg. Unlike most organ systems, which reside entirely within the protective confines of the body cavity, the male reproductive system spans both internal and external territories. A design choice driven by thermal requirements we'll explore shortly. Let's begin with the testes, the paired male gonads, and the primary reproductive organs. These oval structures, each roughly 4 to 5 centimeters in length and 2.5 centimeters in width, serve dual purposes. They produce sperm cells, the male gametes, and synthesize testosterone, the principal male sex hormone. This dual function, both producing reproductive cells and hormones, categorizes the testes as both reproductive organs and endocrine glands. The testes reside in the scrotum, a sac of skin and superficial fascia that hangs outside the body cavity. This external positioning might seem peculiar, even vulnerable, but it serves a crucial function. Sperm production requires a temperature approximately 2 to 3 degrees Celsius below normal body temperature. At 37 degrees, the standard internal temperature, spermatogenesis fails or produces malformed sperm. The scrotum solves this problem by housing the testes outside the body where they remain slightly cooler. The scrotum itself is not merely a passive container. It contains a layer of smooth muscle called the dartos muscle, and associated with each testis, a band of skeletal muscle fibers called the cremaster muscle. These muscles respond to temperature changes. In cold conditions, they contract, pulling the testes closer to the body to conserve warmth. In warm conditions, they relax, allowing the testes to hang lower and dissipate heat. This dynamic temperature regulation occurs continuously, automatically, ensuring optimal conditions for sperm production. Within each testis lies a complex internal architecture. The organ is encased in a tough, fibrous capsule called the tunica albuginea. From the inner surface of this capsule, septa, thin walls of connective tissue, extend inward, dividing the testes into 250 to 300 small compartments called lobules. Each lobule contains 1 to 4 tightly coiled seminiferous tubules, the actual sites of sperm production. If you could uncoil all the seminiferous tubules from both testes and stretch them end to end, they would extend approximately 250 meters, roughly the length of 2.5 football fields. This extraordinary length provides the surface area necessary for continuous, massive sperm production. Between the seminiferous tubules lies loose connective tissue containing Leydig cells, also called interstitial cells. These cells respond to luteinizing hormone from the pituitary gland by producing testosterone. This hormone diffuses into nearby blood vessels and is distributed throughout the body, where it maintains male secondary sexual characteristics, supports sperm production, and influences libido and behavior. The seminiferous tubules themselves are lined with 2 types of cells. Germ cells at various stages of development will become sperm. Sertoli cells, also called sustentacular or nurse cells, support and nourish developing sperm, regulate the environment within the tubule, and form the blood-testis barrier, a selective boundary that protects developing sperm from immune attack and harmful substances. We'll explore spermatogenesis in detail in the next chapter, but for now, know that sperm production begins at the outer edge of the seminiferous tubule and proceeds inward, with mature sperm eventually released into the tubule's central lumen. Once sperm are released into the seminiferous tubules, they are not yet capable of fertilization. They cannot swim effectively, and they lack the molecular machinery necessary to recognize and bind to an egg. These immature sperm must now embark on a journey through a series of ducts and tubules where they will mature and be stored until needed. From the seminiferous tubules, sperm flow into the retestis, a network of short tubules located near the posterior of the testis. From there, they enter 15 to 20 efferent ductules, which convey them out of the testis and into the epididymis. The epididymis is a comma-shaped structure that sits along the posterior border of each testis. Its name, from Greek words meaning upon the twins, refers to its position atop the testis. Despite appearing as a single structure, the epididymis is actually a single, highly coiled duct measuring approximately 6 meters in length when uncoiled. This remarkable length is compacted into a structure only about 4 centimeters long. The epididymis is divided into 3 regions: the head, body, and tail. Sperm enter the head region, where they begin the maturation process. As they travel through the epididymis over a period of 2 to 12 days, they undergo critical changes. They develop the ability to swim in a forward direction rather than merely thrashing about. Their membranes are chemically modified, acquiring proteins that will be essential for fertilization. By the time sperm reach the tail of the epididymis, they are mature and motile, though they remain relatively inactive, conserving energy for their eventual journey. The tail of the epididymis also serves as a storage site. Mature sperm accumulate here, waiting in reserve. They can remain viable in storage for several weeks. If not ejaculated, old sperm are eventually broken down and reabsorbed, their components recycled, while fresh sperm continuously arrive from the testis. From the epididymis, we move to the vas deferens, also called the ductus deferens. This is a muscular tube, approximately 30 to 45 centimeters in length, that transports sperm during ejaculation. The vas deferens begins at the tail of the epididymis, ascends along the posterior testis, and enters the inguinal canal, a passageway through the abdominal wall. It then travels into the pelvic cavity, loops over the urinary bladder, and descends along the bladder's posterior surface. The walls of the vas deferens contain 3 layers of smooth muscle oriented in different directions, giving the duct powerful peristaltic ability. During ejaculation, waves of muscular contraction propel sperm rapidly through the vas deferens. You can actually palpate the vas deferens through the scrotal skin, where it feels like a firm cord. This accessibility makes the vas deferens the target for vasectomy, a surgical sterilization procedure in which a portion of each vas deferens is removed or blocked, preventing sperm from reaching the urethra. Near its end, the vas deferens widens into a region called the ampulla. Here it joins with a duct from the seminal vesicle to form the ejaculatory duct. The 2 ejaculatory ducts, one from each side, are short tubes, approximately 2 centimeters long, that pierce the prostate gland and empty into the urethra. The urethra is the final duct in the male reproductive system, extending from the bladder through the penis to the external urethral opening. In males, the urethra serves dual purposes. It conveys urine during urination and semen during ejaculation, though not simultaneously. The urethra is divided into 3 regions. The prostatic urethra passes through the prostate gland. The membranous urethra passes through the pelvic floor, and the spongy or penile urethra extends through the length of the penis. Along this pathway, 3 sets of accessory glands contribute secretions that mix with sperm to form semen. These glands produce the bulk of seminal fluid, the liquid medium that nourishes, protects, and transports sperm. The seminal vesicles are paired glands located posterior to the bladder. Each is approximately 5 centimeters long and has a convoluted, sac-like shape. The seminal vesicles produce a viscous, yellowish secretion that constitutes approximately 65 to 75% of semen volume. This secretion is rich in fructose, a simple sugar that provides sperm with energy for motility. It also contains prostaglandins, hormone-like substances that may stimulate muscular contractions in the female reproductive tract, helping to propel sperm toward the egg. Additional components include enzymes, vitamin C, and other substances that support sperm viability. The prostate gland is a single, walnut-sized organ that surrounds the prostatic urethra just below the bladder. It consists of 20 to 30 small glands embedded in a matrix of smooth muscle and connective tissue. The prostate contributes approximately 25 to 30% of semen volume. Its secretion is a thin, milky, slightly alkaline fluid containing enzymes, citric acid, and prostate-specific antigen, or PSA, an enzyme that helps liquefy semen after ejaculation. The alkalinity of prostatic fluid is important because it helps neutralize the acidic environment of the vagina, protecting sperm and enhancing their motility. The bulbourethral glands, also called Cowper's glands, are 2 pea-sized structures located inferior to the prostate, near the base of the penis. Each has a duct approximately 2.5 centimeters long that empties into the spongy urethra. During sexual arousal, before ejaculation, the bulbourethral glands secrete a clear, slippery, alkaline mucus. This pre-ejaculatory fluid, sometimes called pre-ejaculate, lubricates the urethra and neutralizes any residual acidity from urine. While it may contain some sperm if a recent ejaculation occurred, its primary purpose is preparation, not sperm transport. The penis is the external male organ responsible for delivering semen into the female reproductive tract. It consists of three cylindrical bodies of erectile tissue, two corpora cavernosa located on the dorsal side, and one corpus spongiosum on the ventral side. The urethra passes through the corpus spongiosum. These bodies are composed of spongy tissue containing numerous vascular spaces. During sexual arousal, these spaces fill with blood, causing the penis to enlarge and become rigid, an erection. The distal end of the corpus spongiosum expands to form the glans penis, the most sensitive region. The skin covering the glans is relatively thin and contains numerous nerve endings. In uncircumcised males, a fold of skin called the foreskin or prepuce covers the glans. At the tip of the glans is the external urethral opening through which both urine and semen exit the body. Erection is controlled by the autonomic nervous system. During sexual arousal, parasympathetic signals cause arteries supplying the erectile tissue to dilate dramatically, increasing blood flow. Simultaneously, compression of veins that normally drain the erectile tissue restricts outflow. Blood accumulates in the vascular spaces, causing expansion and rigidity. After ejaculation, or when arousal subsides, sympathetic signals reverse the process, allowing blood to drain and the penis to return to its flaccid state. The entire male reproductive system, from testes through ducts to accessory glands and penis, functions as an integrated production and delivery system. Sperm are manufactured in the testes, matured and stored in the epididymis and vas deferens, mixed with nutrient-rich and protective fluids from the accessory glands, and expelled through the urethra during ejaculation. This system operates continuously from puberty onward. Unlike the female system, which is cyclical and prepares one gamete per month, the male system produces sperm without pause. A healthy adult male produces approximately 200 to 300 million sperm per day. Over a lifetime, this amounts to trillions of sperm cells, though only a tiny fraction will ever have the opportunity to attempt fertilization. This continuous production strategy reflects a fundamental principle of male reproduction: abundance. Evolution has favored massive overproduction because the challenges facing sperm are immense. The journey from deposition in the vagina to potential fertilization in the fallopian tube is fraught with hazards. Most sperm are lost immediately. Many are malformed or immotile. Others are destroyed by the female immune system or lost in the wrong direction. Of the millions delivered, only a few hundred will reach the vicinity of the egg. With the anatomy mapped, we can now turn our attention to the cellular level and examine the remarkable process by which a single germ cell transforms into millions of specialized sperm cells, each carrying half the genetic material necessary to create a new human being. Spermatogenesis is the process by which diploid germ cells in the testes transform into haploid sperm cells capable of fertilization. It is one of the most precisely orchestrated cellular processes in the human body, requiring approximately 74 days from start to finish and producing millions of new sperm each day from puberty through old age. This process occurs within the seminiferous tubules, those tightly coiled structures that fill the testis. If you were to examine a cross-section of a seminiferous tubule under a microscope, you would see cells at various stages of development arranged in layers, like the concentric rings of a tree. The most immature cells are found at the outer edge, near the basement membrane. As cells mature, they move progressively toward the center, the lumen, where fully formed sperm are eventually released. The journey begins with spermatogonia, diploid stem cells located along the basement membrane of the seminiferous tubule. These cells contain the full complement of 46 chromosomes, 23 pairs, just like other body cells. Spermatogonia are present from fetal development, but they remain inactive until puberty. When puberty begins, triggered by hormonal changes we'll explore later, spermatogonia activate and begin dividing. Spermatogonia undergo mitotic division, standard cell division that produces identical copies, but not all spermatogonia commit to becoming sperm. Some remain as stem cells, dividing to maintain the population and ensure a continuous supply throughout life. Others, called primary spermatocytes, commit to the path of differentiation. They will undergo meiosis and eventually become sperm. Before entering meiosis, primary spermatocytes undergo a period of growth, roughly doubling in size. During this time, they replicate their DNA so that each chromosome consists of two sister chromatids joined at the centromere. With DNA replicated, they are ready for meiosis 1, the first division. Meiosis 1 is a reductional division. Its purpose is to separate homologous chromosomes, reducing the chromosome number from diploid to haploid. Remember that in diploid cells, chromosomes exist in pairs. One chromosome of each pair came from the mother, one from the father. During meiosis 1, these homologous chromosomes pair up, a process called synapsis. While paired, they may exchange segments of DNA in a process called crossing over or genetic recombination. This shuffling of genetic material ensures that each resulting sperm cell carries a unique combination of genes. After recombination, homologous chromosomes separate and move to opposite poles of the cell. The cell then divides, producing two secondary spermatocytes. Each secondary spermatocyte is haploid, containing 23 chromosomes. However, each chromosome still consists of two sister chromatids. A second division, meiosis II, is necessary to separate these chromatids. Meiosis II resembles mitosis. The sister chromatids separate, and the cell divides. Each secondary spermatocyte produces two spermatids, small round haploid cells containing 23 single chromatid chromosomes. From one primary spermatocyte entering meiosis I, 4 spermatids emerge, each genetically unique due to recombination and the random assortment of chromosomes. Spermatids are haploid and contain the correct amount of genetic material, but they do not yet resemble sperm. They are round cells with no specialized structures. The transformation from spermatid to mature sperm, called spermiogenesis, is a remarkable metamorphosis. During spermiogenesis, the spermatid elongates, developing a distinct head and tail. The nucleus condenses, its chromatin compacting tightly, reducing the head to a streamlined shape ideal for swimming through fluids and penetrating barriers. Much of the cytoplasm is discarded, sloughed off as a residual body that will be phagocytized by Sertoli cells. Efficiency, not excess baggage, is the priority. At the anterior end of the head, the Golgi apparatus transforms into a cap-like structure called the acrosome. The acrosome is essentially a large vesicle filled with hydrolytic enzymes, including hyaluronidase and acrosin. These enzymes will be crucial during fertilization for penetrating the protective layers surrounding the egg. Without a functional acrosome, a sperm cannot fertilize an egg, no matter how well it swims. Behind the nucleus, mitochondria arrange themselves in a spiral around the beginning of the tail, forming the midpiece. These mitochondria will provide the ATP necessary to power the tail's movement. Sperm are among the most energy-demanding cells in the body relative to their size. They must swim distances that, proportionally, would be equivalent to a human swimming across an ocean. The mitochondria in the midpiece are the engines for this journey. The tail itself, also called the flagellum, is a long whip-like extension built around a core structure called the axoneme. The axoneme consists of 9 pairs of microtubules arranged in a circle around a central pair, a 9+2 arrangement found in many motile cilia and flagella throughout biology. Motor proteins called dyneins cause adjacent microtubule pairs to slide past each other, creating bending waves that propagate along the tail, propelling the sperm forward. By the end of spermiogenesis, the mature sperm, or spermatozoon, has a distinct structure optimized for a single function: deliver genetic material to an egg. The human sperm cell is approximately 60 micrometers in total length. The head measures about 5 micrometers long and 3 micrometers wide. The tail constitutes the bulk of the length, approximately 50 micrometers. Despite this length, the sperm cell is incredibly small. Millions could fit on the head of a pin. Once spermiogenesis is complete, mature sperm are released from the Sertoli cells into the lumen of the seminiferous tubule, a process called spermiation. They are now free-floating in the tubule, but they are not yet fully mature. They still cannot swim effectively, and they lack the molecular characteristics necessary for fertilization. They must undergo further maturation during their passage through the epididymis, which we'll explore in the next chapter. Throughout spermatogenesis, Sertoli cells play an indispensable role. These large, irregularly shaped cells extend from the basement membrane to the lumen of the seminiferous tubule. They surround developing germ cells, providing structural support, nutrients, and regulatory signals. Sertoli cells form tight junctions with each other, creating the blood-testis barrier. This barrier divides the seminiferous tubule into 2 compartments: a basal compartment near the basement membrane and an adluminal compartment toward the center. The blood-testis barrier is essential for several reasons. Developing germ cells express surface proteins that the immune system would recognize as foreign. After all, they are haploid cells with only half the normal genetic complement, and they undergo extensive modifications during spermatogenesis. If immune cells gained access, they would attack and destroy developing sperm, causing infertility. The barrier prevents this by restricting immune cell entry. The barrier also creates a controlled microenvironment. Sertoli cells regulate what enters the adluminal compartment, ensuring that developing sperm receive appropriate nutrients while being shielded from harmful substances. Sertoli cells also secrete fluid that helps transport sperm toward the epididymis, and they phagocytize defective germ cells and excess cytoplasm shed during spermiogenesis, maintaining cleanliness within the tubule. Sertoli cells respond to hormones: follicle-stimulating hormone, or FSH, from the pituitary gland binds to receptors on Sertoli cells, stimulating them to support spermatogenesis. Without FSH, sperm production declines. Sertoli cells also respond to testosterone, which is present in high concentrations within the testis. Testosterone is essential for completing the later stages of spermatogenesis. When testosterone levels are low, sperm production is impaired. Testosterone is produced by Leydig cells, which reside in the interstitial spaces between seminiferous tubules. Leydig cells respond to luteinizing hormone, or LH, from the pituitary gland. When LH binds to receptors on Leydig cells, it stimulates the synthesis of testosterone from cholesterol through a series of enzymatic reactions. testosterone diffuses into nearby blood vessels and is carried throughout the body, but it is also taken up by Sertoli cells and developing germ cells within the testis, where its local concentration is 50 to 100 times higher than in blood. The entire process of spermatogenesis, from spermatogonium to mature sperm, requires approximately 74 days under optimal conditions. However, sperm are not produced in synchronized batches. Instead, spermatogenesis occurs in waves along the length of each seminiferous tubule. At any given time, different sections of the tubule contain cells at different stages of development. This ensures continuous production. Every day, millions of new sperm complete development and are released into the tubule lumen. A healthy adult male produces approximately 200 to 300 million sperm per day, though this number varies with age, health, and environmental factors. Sperm production is sensitive to temperature, as we noted earlier. Elevated testicular temperature, whether from fever, hot baths, or tight clothing, can impair spermatogenesis. It takes weeks for production to recover after temperature-related damage because cells currently in development must complete their lengthy maturation process, even if compromised. Sperm production is also sensitive to toxins, alcohol, tobacco, certain drugs, environmental pollutants, and exposure to radiation can damage germ cells or disrupt the hormonal regulation of spermatogenesis. Because the process requires more than 2 months, damage may not become apparent immediately. Men who have received chemotherapy or radiation treatment often experience temporary or permanent infertility, depending on the dose and duration of treatment. Despite these vulnerabilities, spermatogenesis is remarkably robust. The continuous production strategy, stem cell population, and large output provide redundancy. Even if production is impaired, reducing daily output by half, hundreds of millions of sperm are still produced daily. Of course, quality matters as much as quantity. Sperm must be properly formed, motile, and carry intact genetic material. Abnormal sperm, lacking heads, having multiple tails, or carrying damaged DNA, are produced in small percentages even under normal conditions. The body's strategy is to overproduce, accepting that many sperm will be defective, but ensuring that vast numbers of healthy sperm are also available. As mature sperm are released into the lumen of the seminiferous tubules, they begin a journey through the male reproductive tract. They are pushed along by fluid, secreted by Sertoli cells, and by gentle contractions of the tubule walls. They flow into the retestis, then through efferent ductules into the epididymis, where the next critical phase of their development will occur. The sperm that enter the epididymis are structurally complete but functionally immature. They must now acquire the capacity to swim effectively and to recognize and fertilize an egg, a process we'll explore next. When sperm leave the seminiferous tubules, they are structurally complete but functionally immature. They possess heads, midpieces, and tails, but they cannot swim properly. If placed in a fluid medium, they would thrash about ineffectively, moving in circles or simply vibrating in place rather than swimming forward. Moreover, their membranes lack certain proteins and surface modifications necessary for recognizing and binding to an egg. In their current state, these sperm are incapable of fertilization. They must undergo a maturation process during their passage through the epididymis. The epididymis, that tightly coiled duct sitting along the posterior edge of the testis, is not merely a passive conduit. It is a site of active maturation where sperm acquire the characteristics essential for fertility. The journey through the epididymis takes between 2 and 12 days, depending on the frequency of ejaculation and other factors. During this time, sperm undergo biochemical and structural changes that transform them from immotile cells into capable swimmers. The epididymal environment is carefully controlled. The epithelial cells lining the duct secrete a complex fluid containing proteins, glycoproteins, ions, and other molecules. This fluid bathes the sperm, and its composition changes along the length of the epididymis, creating different microenvironments in the head, body, and tail regions. These regional differences guide the maturation process in stages. One of the most important changes involves the sperm plasma membrane. As sperm travel through the epididymis, proteins on their surface are modified. Some proteins are added, others are removed, and still others undergo chemical changes such as glycosylation. These modifications are crucial for two reasons. First, they alter the biophysical properties of the membrane, affecting its fluidity and stability. Second, they add or modify receptors that will be necessary for binding to the zona pellucida, the protective glycoprotein shell surrounding the egg. Changes also occur in the sperm's lipid composition. The membrane becomes enriched with cholesterol and certain phospholipids, making it more stable and better able to withstand the chemical challenges it will face in the female reproductive tract. At the same time, membrane proteins redistribute, with some concentrating in specific regions of the head, particularly over the acrosome. This redistribution is part of preparing the sperm for the acrosome reaction, which will occur during fertilization. The development of motility is perhaps the most visible change. Sperm entering the epididymis cannot swim in a straight line. Their flagella beat, but the movement is uncoordinated. As they progress through the epididymis, particularly through the body and tail regions, their motility improves dramatically. By the time they reach the tail of the epididymis, they exhibit progressive motility, swimming forward in relatively straight paths. The exact mechanisms underlying this development are not entirely understood, but they involve changes in the flagellar structure and in the regulatory systems that control flagellar beating. Interestingly, while sperm gain the capacity for motility during epididymal transit, they do not actually swim actively while in the epididymis. The environment there suppresses motility, keeping sperm relatively quiescent. This is adaptive, conserving energy for the journey ahead. Sperm will remain inactive until ejaculation, when they are suddenly exposed to seminal plasma, which activates their motility. The tail of the epididymis serves as the primary storage site for mature sperm. Here, sperm accumulate, waiting for ejaculation. They can remain viable in storage for several weeks, though fertility gradually declines with prolonged storage. The storage environment is maintained at an acidic pH and low oxygen tension, conditions that help preserve sperm by keeping them in a state of low metabolic activity. If sperm are not ejaculated, they eventually age and degenerate. Old sperm are phagocytized by epithelial cells in the epididymis or vas deferens, and their components are recycled. This turnover ensures that the stored sperm population remains relatively fresh. Meanwhile, newly matured sperm continuously arrive from the testis, maintaining the supply. When ejaculation occurs, the process begins with emission. Emission is the movement of sperm and fluids into the urethra, preparing for expulsion. It is triggered by sympathetic nervous system activation during sexual climax. Smooth muscle in the walls of the epididymis, vas deferens, and accessory glands contracts in coordinated waves. Sperm are propelled from the tail of the epididymis into the vas deferens by rhythmic peristaltic contractions. The vas deferens, with its thick muscular walls, generates powerful contractions that move sperm rapidly through its length. Simultaneously, the seminal vesicles contract, emptying their secretions into the ejaculatory ducts. The prostate gland also contracts, adding its secretion. These fluids mix with sperm in the prostatic urethra. The secretion from the seminal vesicles, as mentioned earlier, constitutes the majority of semen volume. It is a thick, yellowish fluid rich in fructose, providing sperm with a readily available energy source. Fructose is metabolized by sperm to produce ATP, which powers flagellar movement. The seminal vesicle secretion also contains prostaglandins, which may stimulate contractions in the female reproductive tract, and proteins that contribute to the initial coagulation of semen. Prostatic secretion is thinner and milky in appearance. It contains enzymes, including prostate-specific antigen, or PSA, and several proteases. After ejaculation, semen initially coagulates, forming a gel-like consistency. This coagulation, caused by proteins from the seminal vesicles, may help retain semen in the vagina. Within 15 to 30 minutes, enzymes from the prostate, particularly PSA, break down the coagulum, liquefying the semen and allowing sperm to swim freely. The slightly alkaline pH of prostatic secretion, typically around 7.2 to 7.8, is important for neutralizing the acidic environment of the vagina, which normally has a pH of 3.8 to 4.5. Vaginal acidity protects against bacterial infections but is hostile to sperm. The alkaline components of semen temporarily raise vaginal pH in the vicinity of the cervix, enhancing sperm survival. The bulbourethral glands contribute a small amount of clear, slippery mucus during sexual arousal before ejaculation. This pre-ejaculatory fluid lubricates the urethra and neutralizes any residual acidity from urine that might be present. It emerges from the urethral opening during arousal and can sometimes be noticed as a clear droplet. While its primary function is preparation, it may contain a small number of sperm, particularly if a recent ejaculation occurred and residual sperm remain in the urethra. Once emission is complete, with sperm and seminal fluids collected in the prostatic and bulbar urethra, expulsion occurs. This is the forceful ejection of semen from the urethra, what is commonly called ejaculation. It involves rhythmic contractions of muscles at the base of the penis, including the bulbospongiosus and ischiocavernosus muscles, as well as muscles of the pelvic floor. These contractions, typically occurring at intervals of about 0.8 seconds, forcefully expel semen through the urethra and out of the body. The volume of a typical ejaculation ranges from 2 to 5 milliliters, roughly 1/2 to 1 teaspoon. This may seem like a small amount, but it contains an enormous number of sperm. A normal sperm count ranges from 15 million to over 200 million sperm per milliliter. Thus, a single ejaculation typically delivers between 40 million and 600 million sperm. The World Health Organization defines a normal ejaculate as containing at least 39 million total sperm, though fertility is possible with lower counts. Not all sperm in an ejaculate are healthy or functional. Even in fertile men, a significant percentage of sperm may be morphologically abnormal, having misshapen heads, multiple tails, or other defects. Additionally, some sperm may be immotile or have poor motility. Normal semen typically contains at least 40% motile sperm, with at least 32% exhibiting progressive motility, meaning they swim forward rather than just in place. The composition of semen is complex. By volume, it is approximately 65 to 75% seminal vesicle secretion, 25 to 30% prostatic fluid, and a small percentage from the bulbourethral glands. Sperm themselves constitute less than 5% of the total volume, typically around 2 to 5%. The remainder is fluid, designed to support and transport sperm. Semen contains a remarkable array of substances. In addition to fructose and prostaglandins, it contains citric acid, enzymes such as acid phosphatase and fibrinolysin, zinc in high concentrations, vitamin C, and small amounts of other vitamins and minerals. It also contains antioxidants that protect sperm DNA from oxidative damage and antimicrobial factors that provide some protection against infections. After ejaculation, semen is deposited in the vagina, typically near the cervix if penetration is deep. The sperm now face an immediate challenge, the hostile vaginal environment. The acidity quickly begins to damage sperm, and the immune system recognizes sperm as foreign cells, deploying white blood cells to attack them. Many sperm are lost within minutes. However, the alkaline components of semen provide temporary protection, and the sperm that are most vigorous and well-formed begin swimming toward the cervix. The cervix, the narrow lower portion of the uterus, serves as a gateway to the upper reproductive tract. Its opening, the external os, leads to the cervical canal, which is normally filled with cervical mucus. Cervical mucus is a hydrogel secreted by glands in the cervical epithelium. Its consistency varies throughout the female menstrual cycle. During most of the cycle, it is thick and viscous, forming a barrier that blocks sperm entry. However, around the time of ovulation, under the influence of estrogen, cervical mucus becomes thin, clear, and stretchy, resembling raw egg white. This fertile type mucus has aligned channels that facilitate sperm passage. Sperm that enter the cervical canal during the fertile period find the environment more favorable. Cervical mucus protects them from vaginal acidity and provides a reservoir where sperm can reside for several days, gradually being released into the uterus. Not all sperm enter the cervix. Many remain in the vagina, where they will be destroyed or expelled. Before sperm can fertilize an egg, they must undergo a final maturation process called capacitation. Capacitation occurs in the female reproductive tract and takes approximately 5 to 6 hours. During this time, sperm undergo further membrane modifications. Cholesterol and certain glycoproteins are removed from the sperm membrane, particularly over the acrosome, making the membrane more fluid and preparing it for the acrosome reaction. Additionally, sperm develop a pattern of motility called hyperactivation, characterized by vigorous, whipping movements of the tail. This type of motility is thought to help sperm break free from the cervical mucus reservoir and penetrate the layers surrounding the egg. The molecular mechanisms of capacitation involve changes in intracellular ion concentrations, particularly calcium and bicarbonate, and alterations in membrane potential. These changes activate signaling pathways that ultimately prepare the sperm for fertilization. Without capacitation, sperm cannot undergo the acrosome reaction or fuse with the egg. From ejaculation through capacitation, sperm are on a timed journey. They remain viable in the female tract for up to 5 days, though their fertilizing ability is highest within the first 24 to 48 hours. The egg, once ovulated, is viable for only about 12 to 24 hours. This creates a narrow window for fertilization, typically from a few days before ovulation to a day after. Timing of intercourse relative to ovulation is critical for conception. The path from testis to potential fertilization is long and arduous for sperm. It takes approximately 74 days to produce a mature sperm, another 2 to 12 days for epididymal maturation, and 5 to 6 hours for capacitation after ejaculation. From millions produced daily, only a tiny fraction will ever be ejaculated, and of those ejaculated, only a minuscule fraction will survive long enough to reach an egg, and only one will successfully fertilize it. This system, seemingly inefficient with its massive overproduction and high attrition, is actually a quality control mechanism. Sperm face a gauntlet of challenges, and only the fastest, strongest, and best formed are likely to reach the egg. This ensures that despite the inevitable production of some defective sperm, healthy sperm have the best chance of achieving fertilization. With sperm production, maturation, and transport understood, we must now shift our attention to hormonal regulation. The entire male reproductive system, from spermatogenesis through ejaculation, is coordinated by a complex interplay of hormones originating in the brain and the testes themselves. This hormonal control ensures that reproduction occurs at the right time and under the right conditions. The male reproductive system does not operate autonomously. Every aspect of its function, from the initiation of sperm production at puberty to the regulation of sexual behavior, is controlled by hormones. These chemical messengers coordinate activity across vast distances, ensuring that the brain, pituitary gland, and testes work in concert to maintain fertility. The hormonal regulation of male reproduction centers on what is called the hypothalamic pituitary gonadal axis, or HPG axis. This is a hierarchical system of control with 3 levels. At the top is the hypothalamus, a region of the brain that monitors the body's internal state and sends signals accordingly. The middle level is the anterior pituitary gland, which sits just below the hypothalamus and responds to its signals by releasing its own hormones. At the bottom level are the testes, which respond to pituitary hormones by producing sperm and testosterone. Let's begin at the top. The hypothalamus contains specialized neurons that secrete gonadotropin-releasing hormone, abbreviated NRH. This small peptide hormone, consisting of only 10 amino acids, is remarkably powerful. It is released in pulses, brief bursts occurring approximately every 90 to 120 minutes. These pulses are critical. Continuous non-pulsatile NRH secretion actually suppresses the reproductive system rather than stimulating it. The pulsatile pattern is necessary for proper function. NRH is secreted into a specialized capillary network called the hypophyseal portal system. This vascular connection allows GnRH to travel directly from the hypothalamus to the anterior pituitary without entering general circulation. When GnRH reaches the anterior pituitary, it binds to receptors on certain cells called gonadotrophs. These cells respond by synthesizing and releasing 2 hormones, follicle-stimulating hormone, or FSH, and luteinizing hormone, or LH. FSH and LH are both glycoprotein hormones, meaning they consist of protein chains with attached carbohydrate groups. They are called gonadotropins because they act on the gonads. Despite their names, which derive from their functions in females, both hormones play essential roles in males as well. FSH enters the bloodstream and travels to the testes, where it binds to receptors on Sertoli cells in the seminiferous tubules. This binding stimulates Sertoli cells to support spermatogenesis. Specifically, FSH promotes the production of androgen binding protein, or ABP, which binds testosterone and concentrates it in the seminiferous tubules where it's needed for sperm development. FSH also stimulates Sertoli cells to secrete various growth factors and signaling molecules that nourish developing germ cells and regulate their progression through spermatogenesis. Without adequate FSH, sperm production declines significantly. Men with FSH deficiency, whether from pituitary damage or genetic conditions, often have severely reduced sperm counts or complete absence of sperm in their semen, a condition called azoospermia. This demonstrates FSH's essential role in male fertility. LH also travels through the bloodstream to the testes, but it targets different cells. LH binds to receptors on Leydig cells, those testosterone-producing cells residing in the spaces between seminiferous tubules. When LH binds to its receptors, it activates a signaling cascade inside the Leydig cell that stimulates the production of testosterone from cholesterol. The synthesis of testosterone involves several enzymatic steps. Cholesterol is converted through intermediate compounds, including pregnenolone, progesterone, and androstenedione, finally yielding testosterone. This process occurs in the smooth endoplasmic reticulum and mitochondria of Leydig cells. LH stimulates the rate-limiting step in this pathway, the conversion of cholesterol to pregnenolone, which is why LH is so critical for testosterone production. Testosterone is a steroid hormone, meaning it is lipid-soluble and can pass through cell membranes easily. Once produced, testosterone diffuses out of Leydig cells and into nearby capillaries, entering the bloodstream. However, much of it is also taken up locally by Sertoli cells and developing germ cells within the testis. The concentration of testosterone inside the testis is 50 to 100 times higher than in the general circulation, creating a local environment optimal for spermatogenesis. Testosterone has numerous effects throughout the body. In the reproductive system, it is essential for maintaining spermatogenesis. Without adequate testosterone, the later stages of sperm development fail, resulting in infertility. Testosterone also maintains the structure and function of the male reproductive tract, including the epididymis, vas deferens, seminal vesicles, and prostate. If testosterone levels drop, these structures atrophy. Beyond reproduction, testosterone is responsible for developing and maintaining male secondary sexual characteristics. At puberty, rising testosterone levels trigger a growth spurt, increase muscle mass and bone density, stimulate growth of facial and body hair, deepen the voice by enlarging the larynx, and promote sebaceous gland activity, which contributes to acne. Testosterone also influences libido, sexual arousal, and aggressive or competitive behaviors, though the relationship between testosterone and behavior is complex and influenced by many factors. In the bloodstream, most testosterone is bound to proteins, particularly sex hormone-binding globulin, or SHBG, and albumin. Only about 2 to 3% circulates in free, unbound form. It is this free testosterone that is biologically active, able to enter cells and exert effects. Once inside a cell, testosterone can act directly by binding to androgen receptors, or it can be converted to other active hormones. In some tissues, the enzyme 5-alpha reductase converts testosterone to dihydrotestosterone, or DHT, a more potent androgen. DHT is particularly important for the development of male external genitalia during fetal development and for prostate growth. In other tissues, testosterone can be converted to estradiol and estrogen by the enzyme aromatase. Yes, males produce estrogen, and it plays important roles in bone health and regulation of the reproductive system. The HPG axis does not operate as a simple one-way street. Instead, it involves feedback loops that maintain hormone levels within appropriate ranges. This is where regulation becomes particularly elegant. Testosterone exerts negative feedback on the hypothalamus and anterior pituitary. When testosterone levels rise, testosterone inhibits NRH secretion from the hypothalamus and also directly suppresses LH release from the pituitary. This negative feedback prevents testosterone levels from rising excessively. Conversely, if testosterone levels drop, the inhibition is lifted, NRH and LH secretion increase, and Leydig cells are stimulated to produce more testosterone. This creates a self-regulating loop that maintains testosterone within a relatively narrow range. FSH secretion is also regulated by feedback, but the signal comes not from testosterone, but from a different hormone called inhibin. Inhibin is a peptide hormone produced by Sertoli cells. Its secretion is stimulated when spermatogenesis is proceeding normally. Inhibin enters the bloodstream, travels to the anterior pituitary, and selectively inhibits FSH secretion without affecting LH. When sperm production is high, inhibin levels rise, FSH secretion decreases, and spermatogenesis slows slightly. When sperm production declines, inhibin levels fall, FSH secretion increases, and spermatogenesis is stimulated. This feedback loop fine-tunes sperm production based on current output. Another hormone, activin, also produced by Sertoli cells, has the opposite effect of inhibin. Activin stimulates FSH secretion. The balance between inhibin and activin helps regulate the sensitivity of the pituitary to GnRH and fine-tune FSH levels. This dual regulation, involving both stimulatory and inhibitory signals, provides more precise control than a single feedback mechanism could achieve. The HPG axis is not active throughout life. During fetal development, it is briefly active, helping to masculinize the developing male fetus. After birth, it becomes quiescent during childhood. Puberty represents the reactivation of the HPG axis, a process that begins around age 9 to 14 inches boys, though timing varies considerably. The trigger for puberty is not entirely understood, but it involves the hypothalamus becoming less sensitive to the inhibitory effects of low testosterone levels. As a result, GnRH pulses begin to increase, initially during sleep, then extending into waking hours. Rising NRH stimulates LH and FSH secretion, which in turn stimulate testosterone production and initiate spermatogenesis. Testosterone levels rise gradually over several years, driving the physical changes of puberty. Once established, the HPG axis remains active throughout adult life in males, unlike females who experience a dramatic cessation of reproductive function at menopause. Males do not have a sudden endpoint. Testosterone levels do decline gradually with age, typically beginning around age 30 and continuing at a rate of about 1 to 2% per year. By age 70, testosterone levels may be 30 to 50% lower than at age 25. This gradual decline, sometimes called andropause or late-onset hypogonadism, can lead to decreased libido, reduced muscle mass, increased body fat, and lower bone density, though many men maintain adequate testosterone levels into old age. Sperm production also declines with age, but it typically continues throughout life. Older men may have lower sperm counts and reduced sperm motility, and the risk of genetic abnormalities in sperm increases. Nevertheless, many men remain fertile into their 70 seconds and beyond, unlike women whose fertility ends sharply at menopause. Factors beyond age can affect the HPG axis. Stress, both physical and psychological, can suppress GnRH secretion, reducing testosterone levels and impairing sperm production. This is mediated by cortisol, a stress hormone that inhibits the reproductive axis. Chronic illness, malnutrition, and extreme exercise can also suppress the axis. Elite endurance athletes and men with eating disorders often have low testosterone and reduced fertility. Obesity affects hormonal balance as well. Adipose tissue contains aromatase, the enzyme that converts testosterone to estrogen. In obese men, increased aromatase activity leads to higher estrogen levels and lower testosterone levels. Elevated estrogen exerts negative feedback on the HPG axis, further suppressing testosterone production. This can create a cycle where obesity leads to hormonal imbalance, which promotes further weight gain. Exogenous hormones, those taken from outside the body, also disrupt the HPG axis. Anabolic steroids, synthetic testosterone derivatives used illicitly to build muscle, flood the body with androgens. This causes negative feedback on the hypothalamus and pituitary, shutting down natural GnRH, LH, and FSH secretion. Without FSH and LH, the testes stop producing testosterone and sperm. The testes actually shrink, a condition called testicular atrophy. Men who use anabolic steroids often become infertile, and even after stopping steroid use, it can take months or years for the HPG axis to recover if it fully recovers at all. Certain medical treatments also affect the axis. Opioid pain medications suppress ENRH secretion, leading to low testosterone and infertility in long-term users. Some psychiatric medications, particularly certain antipsychotics, increase prolactin levels. Prolactin, normally involved in milk production, inhibits GnRH when elevated, suppressing reproduction. The HPG axis can also be manipulated for contraceptive purposes. Male hormonal contraceptives, currently in development, work by administering testosterone and progestins, synthetic progesterone-like hormones. These exert negative feedback on the pituitary, suppressing LH and FSH secretion. Without LH and FSH, the testes stop producing testosterone and sperm. The exogenous testosterone maintains secondary sexual characteristics and libido, but spermatogenesis ceases, resulting in temporary infertility. When treatment stops, the axis recovers and fertility returns, typically within several months. Understanding the HPG axis is crucial for diagnosing and treating male infertility. Blood tests measuring LH, FSH, and testosterone levels can reveal where problems lie. High LH and low testosterone suggest Leydig cell failure. High FSH suggests problems with the seminiferous tubules or Sertoli cells. Low LH and FSH suggest hypothalamic or pituitary dysfunction. These patterns guide treatment. For men with hypogonadism, low testosterone levels, treatment may involve testosterone replacement therapy, or, if fertility is desired, administration of gonadotropins, or GnRH, to stimulate the testes. For men with infertility due to hormonal imbalances, correcting underlying conditions, adjusting medications, or hormone treatments can often restore fertility. The male reproductive system, while often viewed as simpler than the female system, is nevertheless intricately regulated. Hormones coordinate every aspect, from the microscopic process of spermatogenesis to the development of masculine features to the motivation and ability to engage in sexual behavior. The HPG axis, with its hierarchical structure and feedback loops, exemplifies biological control systems, maintaining balance while responding dynamically to changing conditions. With hormonal control understood, we can now turn our attention to the female reproductive system, which operates under similar hormonal principles but with markedly different strategies and outcomes. Where the male system is continuous and relatively constant, the female system is cyclical and dramatically variable, reflecting its dual challenge, producing gametes and preparing to nurture new life. The female reproductive system differs from the male system in fundamental ways. Where the male system produces gametes continuously and focuses primarily on production and delivery, the female system operates in carefully orchestrated cycles, producing typically one gamete per month and preparing the body for the possibility of pregnancy. Where male structures are partly external, all female reproductive organs reside within the protective confines of the pelvic cavity. And perhaps most remarkably, where males produce new gametes throughout reproductive life, females are born with all the eggs they will ever have. Let's begin our exploration with the ovaries, the paired female gonads, and the primary reproductive organs. The ovaries are almond-shaped structures, each approximately 3 to 5 centimeters in length, 1.5 to 3 centimeters in width, and about 1 centimeter thick. They are located on either side of the uterus, one on the left and one on the right, in the upper pelvic cavity. Each ovary is held in position by several ligaments. The ovarian ligament connects it to the uterus, the suspensory ligament anchors it to the pelvic wall, and the broad ligament, a fold of peritoneum, provides additional support. Like the testes, the ovaries serve dual functions. They produce gametes, in this case oocytes or eggs, and they secrete hormones, primarily estrogen and progesterone. This makes the ovaries both reproductive organs and endocrine glands. The surface of the ovary is covered by a layer of simple epithelial cells called the germinal epithelium, though this name is misleading because germ cells do not originate from this layer. Beneath the epithelium is a layer of dense connective tissue called the tunica albuginea. The bulk of the ovary consists of two regions, an outer cortex and inner medulla. The cortex contains thousands of ovarian follicles at various stages of development. Each follicle consists of an oocyte surrounded by one or more layers of support cells. The follicles are embedded in a matrix of connective tissue called the stroma. At birth, a female has approximately 1 million follicles, though this number decreases throughout life. Most follicles will never mature. Instead, they undergo a degenerative process called atresia. The medulla is the inner region of the ovary, containing blood vessels, lymphatic vessels, and nerves that enter through the hilum, the point where the ovary attaches to the suspensory ligament. The medulla provides nourishment and support to the cortex. Moving from the ovaries, we encounter the fallopian tubes, also called uterine tubes or oviducts. These are paired muscular tubes, each approximately 10 to 12 centimeters in length, extending from the sides of the uterus toward the ovaries. Despite their names suggesting a connection, the fallopian tubes do not directly attach to the ovaries. Instead, The end of each tube opens into the pelvic cavity near the ovary. Each fallopian tube is divided into four regions. The infundibulum is the funnel shaped opening near the ovary. It has finger like projections called fimbriae that drape over the ovary. During ovulation, when an oocyte is released from the ovary, the fimbriae sweep it into the tube. The ampulla is the widest and longest section, making up about half the tube's length. This is where fertilization typically occurs. The isthmus is a narrower section near the uterus. Finally, the intramural portion passes through the uterine wall, opening into the uterine cavity. The wall of the fallopian tube consists of three layers. The innermost layer is mucosa lined with ciliated epithelial cells. The cilia beat in waves, creating a current that moves the oocyte toward the uterus. Interspersed among ciliated cells are secretory cells that produce a fluid that nourishes the oocyte and any early embryo. The middle layer is smooth muscle that contracts in peristaltic waves, also helping to propel the oocyte. The outermost layer is serosa, a thin covering of connective tissue. The fallopian tubes serve multiple functions. They capture the oocyte after ovulation, provide the site for fertilization, nourish the oocyte and early embryo, and transport the oocyte or embryo to the uterus. This journey takes approximately 3 to 5 days. At the center of the female reproductive system is the uterus, a hollow muscular organ shaped roughly like an inverted pear. In non-pregnant women, the uterus is about 7.5 centimeters long, 5 centimeters wide at its broadest point, and 2.5 centimeters thick. It is capable of enormous expansion during pregnancy. The uterus is divided into three regions. The fundus is the rounded upper portion above the points where the fallopian tubes enter. The body is the main central portion, making up about 2/3 of the uterus. The cervix is the narrow lower portion that projects into the vagina. The cervix itself is divided into the supravaginal portion above the vagina and the vaginal portion that protrudes into the vaginal canal. The uterine wall consists of three layers. The perimetrium is the outer serous membrane, part of the peritoneum that covers much of the uterus. The myometrium is the thick middle layer of smooth muscle, arranged in multiple layers with fibers running in different directions. During pregnancy, the myometrium stretches dramatically, and during labor, it contracts powerfully to expel the fetus. The endometrium is the inner mucous membrane lining the uterine cavity. The endometrium is perhaps the most dynamic tissue in the body. It consists of two layers, a thin basal layer that remains relatively constant, and a thick functional layer that undergoes dramatic changes during the menstrual cycle. Each month, under hormonal influence, the functional layer thickens, becoming rich in blood vessels and glands, preparing to receive and nourish an embryo. If pregnancy does not occur, this layer is shed during menstruation and then regenerates from the basal layer. The cavity of the uterus is normally a potential space, meaning the anterior and posterior walls are in contact. It is flattened and triangular when viewed from the front, with the fallopian tubes opening at the upper corners and the cervix opening at the lower apex. The cervix warrants particular attention. It serves as both a gateway and a barrier between the uterus and vagina. The cervical canal runs through the center, connecting the uterine cavity to the vaginal cavity. The opening of the cervix into the uterus is called the internal os. The opening into the vagina is the external os. The cervical wall contains numerous glands that secrete cervical mucus. This mucus changes in consistency throughout the menstrual cycle, becoming thin and receptive to sperm around ovulation and thick and impenetrable at other times. The cervix also acts as a protective barrier, normally preventing pathogens from ascending into the uterus. Below the uterus is the vagina, a fibromuscular tube approximately 8 to 10 centimeters in length, though this varies considerably among women and can expand substantially during intercourse and childbirth. The vagina extends from the cervix to the external genitalia, tilting posteriorly at approximately a 90-degree angle to the uterus. The vaginal wall consists of 3 layers. The mucosa is the inner lining, composed of stratified squamous epithelium that can withstand friction. This epithelium lacks glands, but the vagina is kept moist by cervical mucus and by fluid that seeps through the vaginal wall during sexual arousal. The muscularis layer contains smooth muscle arranged in circular and longitudinal patterns, allowing the vagina to expand and contract. The vagina serves multiple functions. It receives the penis and semen during intercourse, providing a pathway for sperm to reach the cervix. It serves as the birth canal through which the infant passes during delivery, and it provides a passageway for menstrual flow to exit the body. The vaginal environment is normally acidic, with a pH of 3.8 to 4.5. This acidity is maintained by lactobacilli, beneficial bacteria that colonize the vagina and ferment glycogen from epithelial cells into lactic acid. The acidic environment protects against many pathogens but is hostile to sperm, which is why seminal fluid contains alkaline buffers. The upper end of the vagina forms a recess around the cervix called the fornix, divided into anterior, posterior, and lateral portions. The posterior fornix is deeper and serves as a reservoir for semen after intercourse, keeping it in close proximity to the cervix. External to the vagina are the external genitalia, collectively called the vulva. These structures include the mons pubis, labia majora, labia minora, clitoris, vestibule, and associated glands. The mons pubis is a rounded elevation of adipose tissue overlying the pubic symphysis. After puberty, it becomes covered with pubic hair. The labia majora are 2 longitudinal folds of skin extending from the mons pubis posteriorly. They are homologous to the scrotum in males, containing adipose tissue and, after puberty, hair on their lateral surfaces. The labia majora enclose and protect the other external structures. The labia minora are 2 smaller folds of skin located internal to the labia majora. They are hairless and contain numerous sebaceous glands. Anteriorly, they join to form a hood of tissue over the clitoris called the prepuce. Posteriorly, they join at the fourchette. The clitoris is a small erectile structure located anterior to the urethral opening. It is homologous to the penis in males and is the primary organ of sexual pleasure in females. The clitoris consists of a glans, body, and 2 crura, roots that extend internally and attach to the pubic arch. Like the penis, the clitoris contains erectile tissue that fills with blood during sexual arousal. The glans clitoris is densely innervated with sensory nerve endings, making it extremely sensitive to stimulation. The vestibule is the area enclosed by the labia minora. It contains several openings. The urethral opening is located approximately 2 to 3 centimeters posterior to the clitoris. In females, unlike males, the urethra is separate from the reproductive tract and serves only to convey urine. The vaginal opening, or vaginal introitus, is located posterior to the urethral opening. In young females who have not engaged in intercourse or strenuous physical activity, the vaginal opening may be partially covered by a membrane called the hymen, though considerable variation exists and the hymen is not a reliable indicator of sexual activity. Also, opening into the vestibule are two pairs of glands. The lesser vestibular glands, also called Skenny's glands, are located on either side of the urethra and secrete mucus that lubricates the vestibule. The greater vestibular glands, also called Bartholin's glands, are located on either side of the vaginal opening and secrete mucus during sexual arousal that facilitates intercourse. The perineum is the diamond-shaped region between the pubic symphysis anteriorly, the coccyx posteriorly, and the ischial tuberosities laterally. It is divided into an anterior urogenital triangle containing the external genitalia and urethra, and a posterior anal triangle containing the anus. The perineum contains muscles that support the pelvic organs and can be stretched or torn during childbirth. While not technically part of the reproductive tract, the mammary glands deserve mention as they are functionally related to reproduction. These modified sweat glands are located in the breasts, which overlie the pectoralis major muscles on the anterior chest wall. Each breast contains 15 to 20 lobes of glandular tissue separated by adipose tissue. Each lobe contains smaller lobules where milk is produced during lactation. From the lobules, lactiferous ducts converge toward the nipple, widening into lactiferous sinuses that store milk before it exits through openings in the nipple. The nipple is a cylindrical projection containing smooth muscle that can cause it to become erect. Surrounding the nipple is the areola, a circular area of pigmented skin containing sebaceous glands that secrete an oily substance to lubricate the nipple during nursing. Breast development begins at puberty under the influence of estrogen and progesterone. The mammary glands undergo further development during pregnancy, preparing for lactation, which we will explore in detail when we discuss birth and postpartum physiology. The position and support of reproductive organs are maintained by several ligaments and sheets of connective tissue. The broad ligament is a fold of peritoneum draping over the uterus, fallopian tubes, and ovaries, dividing the pelvic cavity. The round ligaments extend from the sides of the uterus through the inguinal canal, helping to hold the uterus in an anteverted position, tilted forward over the bladder. The uterosacral ligaments extend from the cervix to the sacrum, providing posterior support. Despite these supports, the pelvic organs can shift position. Weakening of pelvic floor muscles and ligaments, often from childbirth or aging, can lead to pelvic organ prolapse, where the uterus, bladder, or rectum descend into or protrude from the vagina. Blood supply to the female reproductive organs comes primarily from branches of the internal iliac arteries. The ovarian arteries, branching directly from the abdominal aorta, supply the ovaries and parts of the fallopian tubes. The uterine arteries, branches of the internal iliac arteries, supply the uterus and contribute to the blood supply of the ovaries and fallopian tubes. The vaginal arteries supply the vagina. This rich vascular supply is necessary because the uterus and ovaries undergo dramatic changes requiring substantial blood flow. Innervation of the reproductive organs comes from both autonomic and somatic nervous systems. Sympathetic and parasympathetic fibers regulate vascular tone, glandular secretions, and smooth muscle contractions. Sensory nerves provide feedback about touch, pain, and other stimuli. The external genitalia are particularly richly innervated, with dense concentrations of sensory receptors contributing to sexual sensation. One of the most significant differences between male and female reproductive systems is the finite supply of gametes in females. While males produce new sperm continuously, Females are born with all the oocytes they will ever have. During fetal development, millions of oogonia, precursor cells, form in the ovaries. By the fifth month of gestation, approximately 6 to 7 million are present. These begin meiosis, becoming primary oocytes, but then arrest in prophase 1 of meiosis 1. This arrest will last for years or decades. At birth, approximately 1 million oocytes remain, the rest having degenerated. By puberty, only about 400,000 to 500,000 remain. Throughout reproductive life, typically from age 12 to 50, only about 400 to 500 oocytes will actually be ovulated. The rest continue to degenerate through atresia. By menopause, virtually no viable oocytes remain. This means that oocytes age as the woman ages. An oocyte ovulated at age 40 has been arrested in meiosis for more than 40 years. This prolonged arrest increases the risk of chromosomal errors, which is why the incidence of chromosomal abnormalities like Down syndrome increases dramatically with maternal age. A woman in her 22nd has approximately a 1 in 1,500 chance of conceiving a child with Down syndrome. By age 40, the risk is about 1 in 100, and by age 45, it's approximately 1 in 30. The female reproductive system is not merely a collection of organs, but a highly integrated system that operates in monthly cycles. Unlike the relatively constant male system, the female system undergoes dramatic changes every 28 days or so, preparing for the possibility of pregnancy. If pregnancy does not occur, the system resets and begins again. These cyclical changes involve coordinated modifications in the ovaries, where follicles develop and oocytes mature, and in the uterus, where the endometrium thickens and then, if pregnancy doesn't occur, is shed. All of this is orchestrated by hormones that rise and fall in carefully timed patterns. Understanding female reproduction requires understanding these cycles, the hormones that drive them, and the cellular events they control. The anatomical structures we've explored provide the stage, but the menstrual cycle provides the script, directing a monthly drama of preparation, anticipation, and, usually, renewal. We'll turn to the cellular details of oocyte production next, examining the process that begins before birth and continues, one oocyte at a time, throughout reproductive life. Oogenesis, the production of female gametes, follows a dramatically different timeline than spermatogenesis. Where spermatogenesis begins at puberty and continues throughout life, oogenesis begins before birth and proceeds in fits and starts, with most oocytes arrested for years or decades, and only a select few completing their development. Understanding oogenesis requires tracing events from fetal life through ovulation, a span that can exceed 4 decades. The process begins during fetal development. Around the 3rd week of gestation, primordial germ cells, the precursors to all gametes, migrate from the yolk sac to the developing gonads. Once they arrive at the ovaries, these cells differentiate into oogonia, diploid cells containing 46 chromosomes. The oogonia undergo rapid mitotic division, proliferating extensively. By the 5th month of fetal development, the ovaries contain approximately 6 to 7 million oogonia, the peak number that will ever exist. Around this time, the oogonia begin to enter meiosis, the specialized cell division that will ultimately reduce their chromosome number from diploid to haploid. As they enter meiosis, they are now called primary oocytes. However, something remarkable happens. The primary oocytes begin meiosis 1, progressing through the early stages, but then they pause. They arrest in prophase 1, a stage where chromosomes have condensed and homologous pairs have formed, but the cell has not yet divided. This arrest is profound. Primary oocytes remain frozen in prophase 1 from fetal life until they are individually ovulated, which may not occur for decades. An oocyte ovulated at age 40 has been suspended in prophase 1 for more than 40 years. This extended arrest is unique among human cells and has significant implications for fertility and genetic health. Not all oocytes survive until birth. Many undergo atresia, a degenerative process that eliminates them. By birth, only about 1 to 2 million oocytes remain. Atresia continues throughout childhood and adult life. By puberty, approximately 400,000 to 500,000 oocytes survive. The rest have degenerated and been reabsorbed. Each surviving primary oocyte becomes surrounded by a single layer of flattened cells called follicular cells, forming what is called a primordial follicle. The primordial follicle is the basic functional unit of the ovary. It consists of the arrested primary oocyte enclosed by follicular cells, all embedded in the ovarian stroma. Thousands of these tiny structures dot the outer cortex of each ovary. Primordial follicles remain quiescent during childhood. At puberty, hormonal changes activate the ovaries, and each month, a cohort of primordial follicles is recruited to begin development. However, most of these will not complete development. Instead, they will undergo atresia at various stages. Typically, only one follicle per month survives to ovulation. Over a woman's reproductive life, only about 400 to 500 follicles will complete development and release their oocytes. The vast majority, more than 99.9%, will degenerate without ever ovulating. The development of a follicle from primordial to mature stage takes several months and occurs in stages. The first stage is the primary follicle. Activated primordial follicles begin to grow. The follicular cells, now called granulosa cells, change from flattened to cuboidal and begin to proliferate, forming multiple layers around the oocyte. The oocyte itself enlarges, increasing its volume dramatically as it accumulates nutrients, ribosomes, mRNA, and other materials needed for early development after fertilization. As the granulosa cells proliferate, they secrete glycoproteins that form a thick, transparent layer around the oocyte called the zona pellucida. This layer will play crucial roles in fertilization, serving as a binding site for sperm and triggering the acrosome reaction. The zona pellucida remains with the oocyte through ovulation and the early stages of development. Meanwhile, cells in the surrounding ovarian stroma organize around the developing follicle, forming a layer called the theca folliculi. The theca differentiates into two layers: the inner theca interna, which contains endocrine cells that will produce hormones, and the outer theca externa, a fibrous capsule providing structural support. As development continues, the follicle transitions to the secondary stage. Fluid-filled spaces called antral cavities appear among the granulosa cells. These cavities coalesce to form a single large cavity, the antrum, filled with follicular fluid. This fluid is a filtrate of blood plasma containing hormones, growth factors, and nutrients. The oocyte, still surrounded by zona pellucida and layers of granulosa cells, is now pushed to one side of the follicle, sitting on a stalk of granulosa cells called the cumulus operus. The follicle is now called an antral follicle or secondary follicle, and it continues to grow, with the antrum expanding and the follicle bulging from the ovarian surface. At this stage, The follicle is approximately 2 to 10 millimeters in diameter and visible to the naked eye. Each menstrual cycle, approximately 10 to 20 follicles are recruited and begin developing into secondary follicles. However, only one typically becomes dominant and continues development. The others undergo atresia. The selection of the dominant follicle occurs through a competitive process influenced by hormones and local factors. The follicle most sensitive to follicle stimulating hormone, FSH, and producing the most estrogen tends to prevail. The dominant follicle continues growing, eventually reaching 20 to 25 millimeters in diameter, approximately the size of a large grape. It is now called a mature follicle or Graafian follicle. At this stage, it creates a visible bulge on the ovarian surface and is ready for ovulation. The granulosa cells surrounding the oocyte form a distinct corona radiata, and the oocyte itself has reached its full size of approximately 120 micrometers in diameter, making it the largest cell in the human body. Throughout follicular development, hormones play critical roles. FSH stimulates granulosa cell proliferation and the production of estrogen. The granulosa cells contain aromatase enzyme, which converts androgens into estrogen. The androgens are produced by theca interna cells in response to luteinizing hormone, LH. The collaboration between the theca and granulosa cells, called the two-cell, 2-gonadotropin model, is essential for estrogen production. Rising estrogen levels have multiple effects. Estrogen stimulates further growth of the follicle, promotes proliferation of the endometrium in the uterus, and exerts feedback on the hypothalamus and pituitary. Initially, estrogen exerts negative feedback, suppressing FSH secretion. This is why non-dominant follicles undergo atresia. They receive less FSH and cannot continue developing. However, when estrogen levels reach a critical threshold, maintained for a sufficient duration, typically at least 48 hours, the feedback switches from negative to positive. High estrogen now stimulates the release of a surge of LH from the anterior pituitary. This LH surge is the trigger for ovulation. The LH surge has multiple effects on the mature follicle. It causes the primary oocyte, arrested in prophase 1 for years or decades, to finally resume meiosis 1. The cell completes the first meiotic division, but this division is highly asymmetric. One cell, the secondary oocyte, receives virtually all the cytoplasm. The other cell, called the first polar body, is tiny, containing chromosomes but little cytoplasm. The first polar body eventually degenerates and is reabsorbed. The secondary oocyte now contains 23 chromosomes, each consisting of two sister chromatids. It immediately begins meiosis 2, but quickly arrests again, this time in metaphase 2. The oocyte will remain arrested at this stage unless fertilization occurs. If a sperm penetrates the oocyte, meiosis 2 will resume and complete. If fertilization does not occur, the oocyte will degenerate after about 24 hours, still arrested in metaphase 2. The LH surge also initiates a cascade of events leading to ovulation. Enzymes are released that degrade the follicle wall at the site of the bulge. Blood flow to the follicle increases. The follicular wall weakens and bulges further. Prostaglandins and other inflammatory mediators are released. Smooth muscle cells in the ovarian wall contract. Approximately 36 hours after the LH surge begins, the follicle ruptures. Follicular fluid, along with the oocyte, still surrounded by its zona pellucida and corona radiata, is expelled from the ovary into the pelvic cavity. This is ovulation. The fimbriae of the nearby fallopian tube sweep over the ovarian surface, and ciliary action draws the oocyte into the tube. Ovulation is not always a seamless event. Some women experience mild to moderate pain, called Mittelschmerz, German for middle pain, occurring mid-cycle. This pain, felt on one side of the lower abdomen, is thought to result from irritation caused by blood or follicular fluid released during rupture. Ovulation can sometimes cause light spotting. After ovulation, the remnants of the ruptured follicle undergo a remarkable transformation. The granulosa and the caca cells that remain in the ovary do not simply degenerate. Instead, stimulated by LH, they transform into a temporary endocrine structure called the corpus luteum, Latin for yellow body, named for its yellowish color caused by accumulated lipids. The corpus luteum is a highly active endocrine gland. It secretes large amounts of progesterone and moderate amounts of estrogen. These hormones prepare the endometrium for implantation, suppress the development of new follicles, and maintain the uterine lining if pregnancy occurs. If pregnancy does not occur, the corpus luteum has a limited lifespan of approximately 14 days. Without a rescue signal from an implanting embryo, it degenerates into a structure called the corpus albicans, a fibrous scar. As the corpus luteum degenerates, progesterone and estrogen levels plummet. This hormonal withdrawal triggers menstruation, the shedding of the uterine lining, and the cycle begins anew. If pregnancy does occur, the developing embryo secretes a hormone called human chorionic gonadotropin, or HCG, which mimics LH and rescues the corpus luteum, maintaining its function. The corpus luteum continues producing progesterone and estrogen throughout the first trimester of pregnancy, supporting the endometrium until the placenta develops and takes over hormone production. The process of oogenesis, from primordial follicle to ovulated oocyte, involves not just the oocyte itself, but also the surrounding somatic cells. Granulosa and theca cells support oocyte growth, produce hormones, and communicate with the oocyte through gap junctions. The oocyte, in turn, secretes factors that regulate granulosa cell function. This bidirectional communication is essential for proper development. The quality of oocytes declines with age. As women age, the oocytes that have been arrested for decades accumulate damage. The machinery that maintains arrest and will later complete meiosis deteriorates. The mitochondria, which provide energy and were inherited from the woman's own mother, accumulate mutations. The spindle apparatus that separates chromosomes becomes less reliable. These age-related changes lead to increased rates of chromosomal abnormalities, particularly aneuploidy, where the oocyte ends up with the wrong number of chromosomes. Most aneuploid embryos either fail to implant or miscarry early in pregnancy. Some, like those with trisomy 21 Down syndrome, survive but have developmental abnormalities. The rate of chromosomal abnormalities increases exponentially after age 35. While a 25-year-old woman has about a 10% chance that any given oocyte is chromosomally abnormal, a 40-year-old woman has about a 50% chance, and a 45-year-old woman has a 75 to 90% chance. This is the primary reason why fertility declines dramatically with age and why miscarriage rates increase. Eventually, usually around age 50, though with considerable variation, the supply of viable follicles is exhausted. No follicles remain that can respond to FSH. Without follicles, estrogen production ceases. Without estrogen, the menstrual cycle stops. This is menopause, the permanent cessation of menstruation and fertility. The transition to menopause, called perimenopause, typically lasts several years. During this time, cycles become irregular, sometimes skipping months. Eventually, menstruation stops entirely. A woman is considered to have reached menopause after 12 consecutive months without a period. Menopause has significant effects beyond fertility. Estrogen affects many tissues throughout the body. Without it, women experience hot flashes, night sweats, vaginal dryness, mood changes, and accelerated bone loss, potentially leading to osteoporosis. Hormone replacement therapy can alleviate these symptoms by providing exogenous estrogen. Unlike males, who retain fertility into old age even as sperm quality declines, females have a definite endpoint. This difference reflects the distinct challenges of female reproduction. A female not only produces the gamete, but may carry a pregnancy for 9 months and nurse an infant for months or years thereafter. These demands are considerable, and evolution has apparently favored limiting reproduction to earlier years when women are physically better able to handle these challenges. The strategy of arresting oocytes for years or decades, while increasing the risk of chromosomal errors with age, also has advantages. It allows oocytes to grow large, accumulating the nutrients and molecular machinery needed to support early development. Mammalian eggs are among the largest cells in biology, precisely because they must sustain the embryo during its journey through the fallopian tube and early stages of implantation before placental connections form. With oocyte production understood, we can now examine the cyclical process that governs female reproduction, the menstrual cycle, where hormones orchestrate the monthly maturation of a follicle, the preparation of the uterus, and, if fertilization does not occur, the resetting of the system to begin again. The menstrual cycle is one of the most intricate biological rhythms in the human body, a monthly orchestration of hormonal signals and tissue responses that prepares the female body for the possibility of pregnancy. Unlike the relatively constant male reproductive system, the female system operates in repeating cycles, each lasting approximately 28 days, though considerable variation exists, with normal cycles ranging from 21 to 35 days. The cycle involves coordinated changes in 2 sites: the ovaries, where follicles develop and ovulation occurs, and the uterus, where the endometrium prepares to receive an embryo. Because events proceed simultaneously in both locations, the cycle is often described from 2 perspectives: the ovarian cycle and the uterine cycle. These are not separate processes, but different aspects of a unified system. By convention, day 1 of the menstrual cycle is defined as the first day of menstruation, when bleeding begins. This is somewhat arbitrary, since menstruation represents the end of an unsuccessful cycle rather than a true beginning, but it provides a consistent reference point. From this starting point, the cycle divides into phases based on dominant events and hormonal profiles. Let's begin with the ovarian cycle, which divides into 3 phases: the follicular phase, ovulation, and the luteal phase. The follicular phase begins on day 1 and extends until ovulation, typically lasting 14 days, but with considerable variation. This is the phase when follicles develop in the ovary. At the start of the follicular phase, several follicles, perhaps 10 to 20, begin to mature simultaneously. Each contains a primary oocyte arrested in prophase 1 of meiosis, surrounded by layers of granulosa cells. These follicles are responding to FSH from the anterior pituitary. FSH levels are rising because the previous cycle's corpus luteum has degenerated, removing the negative feedback from progesterone and estrogen. With this inhibition lifted, FSH secretion increases, stimulating follicular development. As the follicles grow, their granulosa cells proliferate and begin producing estrogen. The theca cells surrounding the follicles produce androgens in response to LH. The granulosa cells then convert these androgens into estrogen using the enzyme aromatase. This two-cell system requires coordination between granulosa and the CACA cells. Early in the follicular phase, estrogen levels are low and multiple follicles are developing. However, as the phase progresses, one follicle typically becomes dominant. This dominant follicle is more sensitive to FSH, grows faster, and produces more estrogen than the others. As it produces more estrogen, the rising estrogen levels exert negative feedback on the pituitary, reducing FSH secretion. Lower FSH levels create a problem for the non-dominant follicles. They cannot produce enough estrogen without adequate FSH stimulation. Unable to keep up, they undergo atresia, degenerating and being reabsorbed by the ovary. The dominant follicle, however, has accumulated enough FSH receptors and produces enough estrogen that it can continue developing despite declining FSH levels. As the dominant follicle continues growing, it becomes a mature Graafian follicle, bulging from the ovarian surface. Its antrum expands, filled with follicular fluid. The oocyte, surrounded by zona pellucida and corona radiata, sits on a stalk of granulosa cells. By this point, the follicle is producing substantial amounts of estrogen, which is having effects throughout the body. Estrogen stimulates the endometrium to proliferate, which we'll discuss shortly. It increases the production of cervical mucus, making it thin and receptive to sperm. It may enhance libido, subtly encouraging sexual activity around the time of ovulation. And critically, once estrogen reaches a threshold level and maintains it for about 48 hours, it triggers positive feedback. This positive feedback is unusual. For most hormones, elevated levels suppress their own production through negative feedback. But in this unique case, high sustained estrogen stimulates the hypothalamus and pituitary, causing a massive surge in LH secretion, accompanied by a smaller increase in FSH. This LH surge marks the end of the follicular phase and initiates ovulation. The LH surge has profound effects on the mature follicle. First, it causes the oocyte to finally complete meiosis I after being arrested for years or decades. The primary oocyte divides asymmetrically, producing a large secondary oocyte and a tiny first polar body. The secondary oocyte immediately enters meiosis II, but arrests again in metaphase II, where it will remain unless fertilized. Second, the LH surge initiates the physical process of ovulation. Enzymes are activated that weaken the follicle wall. Blood flow increases, raising pressure within the follicle. Inflammatory mediators and prostaglandins are released. The weakened wall bulges further, and approximately 36 hours after the LH surge begins, the follicle ruptures. The oocyte, still surrounded by zona pellucida and corona radiata cells, along with follicular fluid, is expelled into the pelvic cavity. Ovulation is the briefest phase of the cycle, a moment rather than a duration, but it is the pivotal event. The fimbriae of the nearby fallopian tube sweep over the ovary, and ciliary currents draw the oocyte into the tube. The oocyte will remain viable for approximately 12 to 24 hours. If sperm are present in the fallopian tube, fertilization may occur. If not, the oocyte will degenerate. After ovulation, the luteal phase begins. This phase is named for the corpus luteum, the structure that forms from the remnants of the ruptured follicle. The granulosa and the caca cells that remain in the ovary do not degenerate. Instead, they transform under the influence of LH into luteal cells, which are larger, filled with lipid droplets, and specialized for hormone production. The corpus luteum becomes a highly active temporary endocrine gland. It produces large amounts of progesterone and moderate amounts of estrogen. Progesterone is the dominant hormone of the luteal phase, just as estrogen was dominant during the follicular phase. Progesterone has several critical functions. It prepares the endometrium for implantation by stimulating glandular secretions and increasing vascularization. It maintains the endometrium if pregnancy occurs. It suppresses GnRH secretion, preventing new follicles from developing while an embryo might be implanting. And it causes basal body temperature to rise slightly by about 0.5 degrees Celsius, which is why tracking basal body temperature can help identify ovulation. The luteal phase has a relatively fixed duration of approximately 14 days. This consistency contrasts with the follicular phase, which can vary considerably in length. Women with longer or shorter cycles typically have variation in the follicular phase, while the luteal phase remains constant at about 14 days. If fertilization and implantation occur, the developing embryo will secrete human chorionic gonadotropin, HCG, which mimics LH and rescues the corpus luteum, maintaining its function. The corpus luteum continues producing progesterone and estrogen, supporting early pregnancy until the placenta develops and takes over hormone production around 10 to 12 weeks of gestation. If fertilization does not occur or implantation fails, the corpus luteum is not rescued. After approximately 14 days, it begins to degenerate, transforming into a fibrous scar called the corpus albicans. As the corpus luteum degenerates, progesterone and estrogen levels plummet. This sudden hormone withdrawal triggers menstruation, the shedding of the endometrial lining. With low progesterone and estrogen, negative feedback is removed, FSH levels begin rising, and a new cohort of follicles is recruited. The cycle begins anew. Now let's consider the uterine cycle, the concurrent changes occurring in the endometrium. The uterine cycle also divides into phases, traditionally called menstrual, proliferative, and secretory phases, corresponding to events in the ovary. The menstrual phase coincides with the first days of the follicular phase in the ovaries. It begins when declining progesterone and estrogen from the degenerating corpus luteum cause the functional layer of the endometrium to shed. This layer had thickened during the previous cycle in preparation for a potential embryo. Without hormonal support, blood vessels in this layer constrict, depriving tissue of oxygen. Cells die, and the layer begins to detach. Menstrual flow consists of blood, tissue fluid, mucus, and endometrial tissue. The volume varies, typically 20 to 80 milliliters over the course of menstruation, which usually lasts 3 to 7 days. The blood does not clot, thanks to fibrinolytic enzymes released by the endometrium that prevent coagulation. During menstruation, only the functional layer is shed. The basal layer, which lies deeper and has its own blood supply, remains intact. This basal layer will regenerate the functional layer during the next phase. The proliferative phase begins as menstruation ends and extends until ovulation, paralleling the follicular phase in the ovaries. Rising estrogen from developing follicles stimulates the endometrium to regenerate and thicken. The menstrual cycle's complexity requires exquisite hormonal orchestration. Like the male reproductive system, female reproduction is governed by the hypothalamic pituitary gonadal axis, or HPG axis. However, where the male system operates relatively constantly with steady hormone levels maintained by negative feedback, the female system cycles through dramatic hormonal fluctuations in a carefully choreographed monthly pattern involving both negative and positive feedback mechanisms. At the top of the hierarchy sits the hypothalamus, a small region at the base of the brain that serves as a critical link between the nervous system and endocrine system. Specialized neurons in the hypothalamus synthesize and secrete gonadotropin releasing hormone, or GnRH. This small peptide hormone, identical in males and females, is the master regulator initiating the cascade of events controlling reproduction. NRH is not released continuously, but in pulses, discrete bursts occurring approximately every 60 to 90 minutes. The pulsatile pattern is critical. Continuous NRH exposure actually desensitizes the pituitary, suppressing gonadotropin release rather than stimulating it. This principle is exploited therapeutically in treatments for hormone-sensitive cancers and endometriosis, where continuous GnRH analogs shut down the reproductive axis. The frequency and amplitude of GnRH pulses vary across the menstrual cycle, though the changes are subtle. During the follicular phase, pulses occur roughly every 90 minutes. During the luteal phase, pulse frequency slows slightly to every 2 to 4 hours, a change induced by progesterone's effects on the hypothalamus. These variations in pulse pattern help regulate the differential secretion of FSH and LH. NRH is secreted into the hypophyseal portal system, a specialized network of blood vessels connecting the hypothalamus directly to the anterior pituitary gland. This direct vascular link allows GnRH to reach the pituitary at high concentrations without dilution in general circulation. When GnRH binds to receptors on gonadotroph cells in the anterior pituitary, it stimulates synthesis and release of two gonadotropins, follicle-stimulating hormone, or FSH, and luteinizing hormone, or LH. FSH and LH are glycoprotein hormones consisting of protein chains with attached carbohydrate groups. They share a common alpha subunit, but have distinct beta subunits that confer specificity. Despite their names derived from early observations in female animals, both hormones have critical functions in females and males, though their specific actions differ between sexes. In females, FSH travels through the bloodstream to the ovaries, where it binds to receptors on granulosa cells surrounding developing follicles. FSH is the primary hormone driving follicular development during the follicular phase. It stimulates granulosa cells to proliferate, increasing the cell layers surrounding the oocyte. It promotes the development of the antrum, the fluid-filled cavity within the follicle, and critically, it stimulates granulosa cells to express aromatase, the enzyme that converts androgens to estrogen. Without adequate FSH, follicles cannot develop properly. Women with FSH deficiency, whether from pituitary damage or certain genetic conditions, experience absent or irregular menstrual cycles and infertility. Conversely, FSH levels rise dramatically during menopause when ovarian follicles are depleted and can no longer respond, removing negative feedback. LH also targets the ovaries, but acts on different cells. In the developing follicle, LH binds to receptors on the CACA cells, stimulating them to produce androgens, primarily androstenedione and testosterone, These androgens diffuse to adjacent granulosa cells, where aromatase converts them to estrogen, primarily estradiol. This two-cell, two-gonadotropin system requires cooperation between the theca and granulosa cells, with LH and FSH working in tandem. As the menstrual cycle progresses, granulosa cells acquire LH receptors, making them responsive to LH as well as FSH. This acquisition is stimulated by FSH and occurs in the late follicular phase. When the LH surge occurs at mid-cycle, the now LH-responsive granulosa cells undergo dramatic changes, completing the final stages of follicular maturation and triggering ovulation. Let's trace hormone levels through a complete menstrual cycle to see how the system operates. At the start of the cycle, day 1 defined as the first day of menstruation, FSH levels are rising. This rise began late in the previous cycle, when the degenerating corpus luteum stopped producing progesterone and estrogen, removing negative feedback. With inhibition lifted, FSH increases, recruiting a cohort of follicles to begin developing. LH levels are also relatively low but begin rising as well. The ratio of FSH to LH during early follicular phase favors FSH, which is appropriate since follicle development is the priority. As follicles develop over the next week, they produce increasing amounts of estrogen. Initially, estrogen exerts negative feedback on the hypothalamus and pituitary. This negative feedback is dose-dependent. Moderate estrogen levels suppress GnRH pulses and reduce FSH and LH secretion. This suppression serves an important function. It creates competition among developing follicles. As FSH levels decline due to rising estrogen's negative feedback, follicles must compete for the diminishing FSH supply. The follicle that is most sensitive to FSH has accumulated the most FSH receptors and produces the most estrogen gains an advantage. This becomes the dominant follicle. The others, unable to obtain sufficient FSH stimulation, undergo atresia. The dominant follicle continues growing and producing more estrogen. Estrogen levels rise steadily throughout the follicular phase. Around days 10 to 12, estrogen levels begin rising sharply as the dominant follicle approaches maturity. Here, the system performs a remarkable reversal. When estrogen reaches a threshold level, approximately 200 picograms per milliliter, and maintains this level for about 48 hours, the negative feedback switches to positive. High sustained estrogen now stimulates NRH secretion and sensitizes gonadotrophs to NRH. The result is a massive surge in LH secretion, with levels increasing tenfold or more over 24 to 48 hours. FSH also surges, though less dramatically. This mid-cycle LH surge is the trigger for ovulation. It has multiple effects on the mature follicle. It causes the primary oocyte to complete meiosis I, forming a secondary oocyte and first polar body. It initiates changes in granulosa and the theca cells that will transform them into the corpus luteum. It activates enzymes that weaken the follicle wall, and approximately 36 hours after the surge begins, it causes follicle rupture and ovulation. The positive feedback mechanism triggering the LH surge is unusual in endocrine regulation. Most hormone systems use negative feedback exclusively to maintain homeostasis. The positive feedback in the female reproductive system creates a dramatic, transient deviation from steady state necessary to trigger the discrete event of ovulation. After ovulation, hormone patterns shift dramatically. LH and FSH levels decline from their surge peaks. The ruptured follicle transforms into the corpus luteum under the influence of the residual LH. The corpus luteum becomes the dominant endocrine structure of the luteal phase, producing large amounts of progesterone and moderate amounts of estrogen. Progesterone levels rise steadily throughout the luteal phase, peaking around days 20 to 22, about 1 week after ovulation. Progesterone is now the dominant hormone, and its effects are profound. It transforms the endometrium from a proliferative to a secretory state. It causes basal body temperature to rise by about 0.5 degrees Celsius, which is why tracking basal body temperature can identify ovulation retrospectively, and it exerts powerful negative feedback on the hypothalamus and pituitary. Progesterone slows GnRH pulse frequency and suppresses LH and FSH secretion. This makes biological sense with the endometrium preparing for potential implantation. Developing new follicles would be counterproductive. The suppression ensures that energy and resources focus on supporting a potential pregnancy rather than initiating a new cycle. Estrogen levels also rise during the luteal phase, though they remain lower than the preovulatory peak. Estrogen and progesterone together maintain the endometrium and provide negative feedback to the hypothalamus and pituitary, keeping FSH and LH suppressed. If pregnancy occurs, the implanting embryo begins secreting human chorionic gonadotropin, or HCG, around 6 to 7 days after fertilization. HCG is structurally similar to LH and binds to LH receptors on the corpus luteum. This rescues the corpus luteum from degeneration, maintaining its production of progesterone and estrogen. The corpus luteum continues functioning throughout the first trimester, supporting the endometrium until the placenta develops and assumes hormone production. If pregnancy does not occur, the corpus luteum has a fixed lifespan of approximately 14 days. In the absence of HCG rescue signals, it begins degenerating around day 28 of the cycle. Progesterone and estrogen levels plummet rapidly. This sudden hormone withdrawal has multiple effects. The spiral arteries supplying the endometrial functional layer constrict, causing ischemia and tissue death. The functional layer begins to detach, initiating menstruation. Simultaneously, the removal of negative feedback allows FSH and LH to begin rising again. Rising FSH recruits a new cohort of follicles, and a new cycle begins. The cycle has come full circle, ready to repeat its monthly preparation for pregnancy. Beyond FSH, LH, estrogen, and progesterone, several other hormones modulate the reproductive system. Inhibin, a protein hormone produced by granulosa cells, selectively suppresses FSH secretion without affecting LH. Inhibin levels rise when follicles are actively developing, providing feedback about ovarian activity. If follicular development is impaired, inhibin levels fall and FSH rises, potentially compensating by stimulating more follicles. Activin, also produced by granulosa cells, has the opposite effect, stimulating FSH secretion. The balance between inhibin and activin fine-tunes FSH levels throughout the cycle. Anti-Mullerian hormone, or AMH, produced by small preantral and small antral follicles, inhibits the recruitment of primordial follicles and decreases sensitivity to FSH. AMH levels reflect the ovarian reserve, the number of remaining follicles. As women age and follicle numbers decline, AMH levels decrease, which is why AMH testing is used clinically to assess fertility potential. Prolactin, the hormone primarily associated with milk production, can affect reproduction when levels are abnormally elevated, a condition called hyperprolactinemia. High prolactin suppresses GnRH secretion, disrupting the menstrual cycle and causing infertility. This can occur with prolactin secreting pituitary tumors or as a side effect of certain medications, particularly some antipsychotics. Androgens also play roles in female reproduction, though at much lower levels than in males. The ovaries and adrenal glands produce small amounts of testosterone and other androgens. These contribute to libido and may influence follicular development. However, excess androgens, as seen in PCOS, disrupt normal cycles, causing irregular menstruation and infertility. The hypothalamic pituitary ovarian axis does not operate in isolation. It is influenced by signals from throughout the body. The thyroid gland, for instance, affects reproduction. Both hyperthyroidism, excess thyroid hormone, and hypothyroidism, insufficient thyroid hormone, can disrupt menstrual cycles and impair fertility. Thyroid hormones influence the metabolism of reproductive hormones and affect ovarian response to gonadotropins. Stress, both physical and psychological, profoundly affects the reproductive axis. Stress activates the hypothalamic pituitary adrenal axis, increasing cortisol production. Cortisol suppresses GnRH secretion, disrupting the reproductive cycle. Women experiencing chronic stress often have irregular cycles or amenorrhea. This is an adaptive response in times of perceived threat or scarcity. Reproduction is deprioritized in favor of immediate survival. Nutrition and body composition also affect hormonal balance. Leptin, a hormone produced by adipose tissue, signals nutritional status to the hypothalamus. Adequate leptin levels are necessary for normal GnRH secretion. When body fat percentage drops too low, leptin levels fall, GnRH secretion is suppressed, and menstruation ceases. This is why female athletes and women with anorexia often experience amenorrhea. Conversely, obesity leads to elevated leptin, but also leptin resistance, and to increased estrogen production by adipose tissue, both of which can disrupt normal cycling. The HPG axis undergoes dramatic changes at different life stages. During childhood, the axis is quiescent. NRH neurons are active, but the hypothalamus is extremely sensitive to the negative feedback effects of even minimal sex steroid levels, keeping the system suppressed. Puberty represents the reactivation of the HPG axis. The trigger for puberty is not entirely understood, but it involves decreased sensitivity to negative feedback. NRH pulses begin to increase, initially during sleep, then extending into waking hours. As NRH increases, FSH and LH secretion rises, stimulating the ovaries to produce estrogen. Estrogen drives the physical changes of puberty: breast development, widening of hips, growth of pubic and axillary hair, and acceleration of growth. Eventually, estrogen reaches levels sufficient to trigger the positive feedback mechanism, producing the first ovulatory LH surge. Menarche, the first menstrual period, typically occurs 2 to 3 years after the onset of puberty. Early menstrual cycles are often irregular and anovulatory. It can take months to years for the positive feedback mechanism to become fully functional and for cycles to stabilize into regular ovulatory patterns. At the other end of reproductive life is menopause, the permanent cessation of menstruation. Menopause occurs when the ovarian follicle supply is exhausted. With no follicles remaining to respond to FSH, estrogen production ceases. Without estrogen, The positive feedback mechanism cannot trigger LH surges, ovulation stops, and menstruation ends. The transition to menopause, perimenopause, typically begins in the mid 40 seconds and lasts several years. Follicles decline in number and quality. Cycles become irregular, sometimes occurring more frequently, sometimes less. Some cycles are anovulatory. FSH levels begin rising as the ovaries become less responsive, requiring more stimulation to produce adequate estrogen. Eventually, follicles are depleted, estrogen drops to very low levels, and FSH and LH rise markedly, remaining elevated thereafter. Postmenopausal women have FSH levels 10 to 20 times higher than during reproductive years, reflecting the complete loss of negative feedback from the ovaries. These high gonadotropin levels cannot stimulate follicular development because no follicles remain. The hormonal changes of menopause have widespread effects. Estrogen affects many tissues beyond the reproductive system. Without it, women experience vasomotor symptoms like hot flashes and night sweats, vaginal atrophy and dryness, accelerated bone loss, increasing osteoporosis risk, changes in cholesterol metabolism, increasing cardiovascular disease risk, and various mood and cognitive changes. Hormone replacement therapy with estrogen, often combined with progesterone to protect the endometrium from unopposed estrogen stimulation, can alleviate these symptoms. However, hormone therapy carries risks, including increased incidence of certain cancers and cardiovascular events, so treatment decisions must weigh benefits against risks for each individual. The HPG axis can be manipulated for therapeutic purposes. Oral contraceptives, commonly called birth control pills, contain synthetic estrogen and progestin, a progesterone-like compound. These exogenous hormones exert negative feedback on the hypothalamus and pituitary, suppressing FSH and LH secretion. Without gonadotropin stimulation, follicles do not develop, ovulation does not occur, and pregnancy is prevented. The typical oral contraceptive regimen involves 21 days of active hormone pills, followed by 7 days of placebo pills. During the placebo week, hormone levels drop, triggering withdrawal bleeding that resembles menstruation, though it is not true menstruation since no ovulation occurred. When active pills resume, hormone levels rise again, suppressing the axis. Other hormonal contraceptives work through similar mechanisms. Contraceptive patches deliver hormones through the skin. Vaginal rings release hormones locally. Injectable progestins provide longer-lasting suppression. Implantable rods release progestins slowly over several years. All work by suppressing ovulation through negative feedback. Some contraceptives contain only progestin without estrogen. These work partly by suppressing ovulation, but also by thickening cervical mucus to block sperm passage and by altering the endometrium to prevent implantation. Progestin-only methods are useful for women who cannot tolerate estrogen. Hormonal treatments are also used for infertility. Women who do not ovulate regularly may be treated with clomiphene citrate, a medication that blocks estrogen receptors in the hypothalamus and pituitary. By blocking negative feedback, clomiphene causes FSH and LH to rise, stimulating follicular development. Many women treated with clomiphene successfully ovulate and conceive. For women who do not respond to clomiphene, injectable gonadotropins can directly stimulate the ovaries. These medications contain FSH, LH, or both, bypassing the hypothalamus and pituitary to directly stimulate follicular development. Gonadotropin treatment requires careful monitoring because it can stimulate multiple follicles simultaneously, increasing the risk of multiple pregnancies. In vitro fertilization, or IVF, uses hormonal manipulation extensively. Women undergoing IVF first receive GnRH agonists or antagonists to suppress their natural cycle, preventing spontaneous ovulation. Then, high doses of gonadotropins stimulate multiple follicles to develop simultaneously. When follicles reach appropriate size, an injection of HCG triggers final maturation. Oocytes are then retrieved surgically, fertilized in the laboratory, and resulting embryos are transferred to the uterus. Understanding the HPG axis also helps diagnose reproductive disorders. Blood tests measuring FSH, LH, estrogen, and progesterone at specific cycle points reveal where problems lie. High FSH with low estrogen suggests ovarian failure. The ovaries cannot respond to stimulation. Low FSH and LH suggest hypothalamic or pituitary dysfunction. The signal to the ovaries is absent or inadequate. Normal hormones with an ovulation might suggest PCOS or other ovarian dysfunction. The female reproductive system's hormonal control is far more complex than the male system's relatively steady state. The monthly cycling through dramatically different hormonal profiles, the switch from negative to positive feedback at mid-cycle, and the precise timing required for all events to align properly make female reproduction a remarkable example of biological coordination. Yet this complexity also creates vulnerability. Many factors can disrupt the delicate balance. The system is sensitive to stress, nutrition, body composition, exercise, illness, and psychological state. Small disruptions can cascade into menstrual irregularities or infertility. But the system is also robust, with built-in redundancies and compensatory mechanisms that usually maintain function even when challenged. With the hormonal orchestration of the female reproductive system understood, we can now turn to the act that brings male and female gametes together, sexual intercourse. This behavior, driven by biology but also influenced by psychology and emotion, is the gateway through which sperm begin their journey toward a waiting egg. Sexual intercourse is the biological mechanism by which sperm are delivered to the female reproductive tract, bringing male and female gametes into proximity. While reproduction can occur through artificial means, natural conception requires this physical union. The act itself involves coordinated physiological responses in both partners, responses that are hardwired into our nervous system, yet also influenced by psychology, emotion, and context. The sexual response in both males and females follows a pattern that can be divided into phases: arousal, plateau, orgasm, and resolution. These phases involve changes in heart rate, blood pressure, muscle tension, and genital blood flow, all coordinated by the autonomic nervous system and mediated by complex interactions between neurological and vascular systems. Sexual arousal begins in the brain, triggered by sensory stimuli, visual, auditory, tactile, olfactory, as well as by thoughts, memories, and emotions. The brain's response involves multiple regions. The limbic system processes emotional content, the hypothalamus coordinates physiological responses, and the cerebral cortex processes conscious thoughts and fantasies. When arousal occurs, the brain sends signals through the spinal cord to the genitals, initiating physiological changes. In males, the primary physiological response is erection. This process is controlled primarily by the parasympathetic nervous system. When arousal signals travel from the brain down the spinal cord, parasympathetic nerve fibers release neurotransmitters, particularly nitric oxide, that cause the arteries supplying the erectile tissue of the penis to dilate dramatically. The penis contains three cylinders of erectile tissue, two corpora cavernosa on the dorsal side and one corpus spongiosum on the ventral side, surrounding the urethra. These tissues consist of a sponge-like network of vascular spaces called sinusoids. Normally, these spaces contain minimal blood, and the penis remains flaccid. During arousal, arterial dilation increases blood flow into the sinusoids by 20 to 40 times normal. The sinusoids fill with blood, expanding. As they expand, they compress the veins that normally drain blood from the penis, reducing outflow. This combination of increased inflow and decreased outflow causes blood to accumulate, and the erectile tissue becomes engorged and rigid. The penis enlarges and stiffens, achieving erection. The degree of rigidity is remarkable. The intracavernous pressure during full erection can exceed systolic blood pressure. This rigidity is necessary for vaginal penetration. Without adequate erection, intercourse cannot occur, a condition called erectile dysfunction. Maintaining erection requires sustained parasympathetic activity. Psychological factors, stress, anxiety, or distraction can shift the balance towards sympathetic activity, which causes the arteries to constrict and the erection to subside. This is why psychological state significantly affects male sexual function. In females, arousal produces several physiological changes. Like male erection, female genital arousal involves increased blood flow to erectile tissues. The clitoris contains erectile tissue homologous to the penile corpora cavernosa. During arousal, this tissue engorges with blood, causing the clitoris to swell and become erect, though less visibly than the penis. Blood flow also increases to the vaginal walls and labia. The vaginal walls become engorged, and the increased pressure causes fluid to pass through the vaginal epithelium, a process called transudation. This fluid lubricates the vagina, facilitating penetration and reducing friction during intercourse. Lubrication typically begins within 10 to 30 seconds of effective stimulation. The greater vestibular glands, or Bartholin's glands, also secrete mucus during arousal, adding to lubrication. The labia minora swell with blood and darken in color. The vagina lengthens and expands, particularly in its inner two-thirds, a process called tenting. This expansion creates space to accommodate the penis and pools semen near the cervix after ejaculation. As arousal continues and intensifies, both males and females enter the plateau phase. Physiological changes intensify. Heart rate, blood pressure, and breathing rate increase. Muscle tension throughout the body rises. Skin may flush, particularly on the chest and face. These systemic changes reflect widespread autonomic nervous system activation. In males, the testes elevate closer to the body as the cremaster muscles contract. The Cowper's glands release pre-ejaculatory fluid, which appears at the urethral opening. The penis reaches maximum engorgement, and the glands may deepen in color. In females, the outer third of the vagina becomes particularly engorged, forming what is called the orgasmic platform. This narrows the vaginal opening, increasing friction during intercourse. The uterus elevates, further expanding the inner vagina. The clitoris retracts under its hood, becoming less accessible to direct stimulation, but still responding to indirect stimulation through movement of surrounding tissues. With continued stimulation, the plateau phase builds toward orgasm. Orgasm is a brief, intensely pleasurable experience involving rhythmic muscular contractions, a sense of release, and altered consciousness. Despite subjective similarities between male and female orgasm, the underlying physiology differs. In males, orgasm typically occurs simultaneously with ejaculation, though these are technically separate processes controlled by different nervous system mechanisms. Ejaculation consists of two phases, emission and expulsion, which we discussed earlier. Emission involves the movement of sperm and seminal fluids into the urethra. Smooth muscle in the epididymis, vas deferens, seminal vesicles, and prostate contracts rhythmically, propelling sperm and fluids into the prostatic urethra. The internal urethral sphincter closes, preventing semen from entering the bladder and urine from mixing with semen. This creates a point of no return once emission begins. Ejaculation becomes inevitable. Expulsion immediately follows emission. Skeletal muscles at the base of the penis, particularly the bulbospongiosus and ischiocavernosus muscles, contract rhythmically at approximately 0.8-second intervals. These contractions forcefully expel semen through the urethra. The first few contractions are most forceful, projecting semen with some velocity. Subsequent contractions are weaker. The male orgasmic experience coincides with these contractions and involves intense pleasure, altered awareness, and involuntary vocalizations or body movements. After ejaculation, males enter a refractory period during which they cannot achieve another erection or orgasm. This period varies from minutes to hours depending on age, health, and other factors, typically lengthening with age. Female orgasm involves rhythmic contractions of the uterus, vaginal walls, and pelvic floor muscles, occurring at similar intervals to male orgasmic contractions, approximately 0.8 seconds apart. These contractions are involuntary, though women can learn to augment them somewhat through pelvic floor muscle control. The uterine contractions begin at the fundus and proceed toward the cervix, though they are much weaker than labor contractions. Whether these contractions help transport sperm toward the fallopian tubes is debated. Some research suggests a potential assist, but sperm are capable of reaching the tubes without female orgasm. Female orgasm is also accompanied by intense pleasure, altered consciousness, involuntary body movements, and often vocalizations. Unlike males, females do not have a refractory period and are capable of multiple orgasms in rapid succession if stimulation continues. After orgasm, both males and females enter the resolution phase, where physiological changes gradually reverse. Blood drains from engorged tissues, heart rate and blood pressure return to baseline, muscle tension dissipates, and a sense of relaxation and sometimes drowsiness sets in. For conception to occur, sperm must be deposited in the vagina, ideally near the cervix. The volume of semen, typically 2 to 5 milliliters, pools in the posterior fornix, the deep recess behind the cervix. This positioning keeps sperm close to the cervical opening, maximizing the number that can enter. The sperm now face an immediately hostile environment. The vagina maintains an acidic pH of 3.8 to 4.5, maintained by lactobacilli bacteria that ferment glycogen into lactic acid. This acidity protects against pathogenic bacteria but is harmful to sperm. Sperm exposed to vaginal acidity begin to die within minutes. However, the semen provides temporary protection. Its alkaline components, particularly from prostatic secretion, neutralize acidity in the immediate vicinity, This creates a brief window during which viable sperm can swim toward the cervix. The female immune system also poses a challenge. Sperm are recognized as foreign cells, and white blood cells in the vagina and cervix begin attacking them. Macrophages and neutrophils phagocytize sperm. Of the millions deposited, many are destroyed within the first hour. The sperm that survive are the fastest and most vigorous. They begin swimming toward the cervix, guided by chemical gradients and thermotaxis, the tendency to swim toward slightly warmer temperatures in the upper reproductive tract. The cervix presents the next barrier. The cervical canal is normally filled with cervical mucus, which varies in consistency throughout the menstrual cycle. During most of the cycle, the mucus is thick, viscous, and relatively impenetrable, forming a barrier that blocks sperm entry. However, around ovulation, under the influence of high estrogen levels, cervical mucus undergoes dramatic changes. It becomes thin, clear, and watery, with a consistency resembling raw egg white. More importantly, its molecular structure changes. The mucin molecules align in parallel strands, creating channels approximately 5 micrometers wide, just wide enough for sperm to pass through in single file. This fertile type mucus facilitates sperm passage in multiple ways. It provides a more neutral pH, protecting sperm from vaginal acidity. Its channels guide sperm in the correct direction. It filters out abnormal sperm, those that are too large, misshapen, or swim poorly, while allowing normal sperm through. And it can store sperm for several days, releasing them gradually into the uterus. Sperm that successfully enter the cervix encounter a more hospitable environment. The mucus provides nutrition and protection. However, they must still navigate the cervical canal, which is approximately 2 to 3 centimeters long. This may seem short, but at a sperm scale, it represents a substantial journey. Sperm swim at approximately 1 to 4 millimeters per minute, so traversing the cervix takes 30 minutes to an hour. Once through the cervix, sperm enter the uterine cavity. Here, the environment is more neutral, and muscular contractions in the uterine walls may assist sperm transport. However, the uterus also contains immune cells that attack sperm. Many more sperm are lost here. The surviving sperm must reach the fallopian tubes. The uterine cavity is triangular, approximately 7 to 8 centimeters from cervix to fundus. Sperm must swim this distance and then locate one of the 2 fallopian tube openings, each only about 1 millimeter in diameter, located at the upper corners of the uterus. Of the millions of sperm originally deposited, only thousands make it into the uterus, and only hundreds reach the fallopian tubes. This dramatic attrition reflects the multiple challenges sperm face and the body's strategy of using competition to ensure that only the most capable sperm approach the egg. Sperm are also distributed between the 2 fallopian tubes. If an egg has been released from one ovary, only sperm entering that tube have a chance of fertilization. Sperm entering the opposite tube will find no egg. The fallopian tube is approximately 10 to 12 centimeters long, divided into the intramural portion passing through the uterine wall, the isthmus, the ampulla, and the infundibulum with its fimbriae. Fertilization typically occurs in the ampulla, the widest section, so sperm must travel most of the tube's length. The journey through the fallopian tube is assisted by muscular contractions and by currents created by ciliated cells lining the tube. However, these forces primarily move in the opposite direction, transporting the egg toward the uterus. Sperm must swim against this current, which slows their progress, but may also serve as additional selection pressure. Before sperm can fertilize an egg, they must undergo capacitation, the final maturation process that occurs in the female reproductive tract. Capacitation involves removal of glycoproteins and cholesterol from the sperm plasma membrane, particularly over the acrosome. These surface modifications are necessary for the acrosome reaction, which will allow the sperm to penetrate the egg's protective layers. Capacitation also involves changes in sperm motility. Capacitated sperm develop hyperactivated motility, characterized by vigorous, whip-like tail movements rather than the smooth, progressive motility of uncapacitated sperm. This hyperactivation helps sperm break free from reservoirs in cervical mucus and may assist in penetrating the layers surrounding the egg. The molecular mechanisms of capacitation involve influx of calcium and bicarbonate ions, changes in membrane potential, increased cyclic AMP levels, and protein phosphorylation cascades. These changes require approximately 5 to 6 hours in the female tract. Sperm can survive in the female reproductive tract for up to 5 days, though their fertilizing capacity is highest during the first 48 hours. They reside in crypts in the cervical mucus and in the isthmus of the fallopian tube, slowly being released and traveling toward the ampulla. The egg, once ovulated, is viable for only 12 to 24 hours. This creates a fertile window of approximately 5 to 6 days, the 5 days before ovulation, during which sperm can survive waiting for the egg, and the day of ovulation itself. Intercourse outside this window is unlikely to result in pregnancy. Timing of intercourse relative to ovulation significantly affects conception probability. The highest pregnancy rates occur when intercourse happens in the 2 days before ovulation. Intercourse on the day of ovulation is also effective, but pregnancy rates decline if intercourse occurs 3 to 5 days before ovulation, as fewer sperm survive to the time of fertilization. Women attempting to conceive can use various methods to identify their fertile window, tracking cervical mucus changes, monitoring basal body temperature shifts, using ovulation prediction kits that detect the LH surge, or tracking menstrual cycle patterns. The journey from ejaculation to potential fertilization is arduous for sperm. From the hundreds of millions deposited in the vagina, only a few hundred reach the vicinity of the egg. This extreme attrition, while seemingly wasteful, serves quality control, ensuring that the sperm that ultimately encounters the egg is likely to be healthy, well-formed, and carrying intact genetic material. With sperm having completed their journey and arrived in the fallopian tube, and with an egg released from the ovary and captured by the fimbriae, the stage is set for fertilization, the union of gametes that will create a new genetically unique individual. Fertilization is the process by which a sperm and an egg fuse to create a single cell called a zygote, the beginning of a new individual. This event typically occurs in the ampulla of the fallopian tube, the widest section located about 2/3 of the way from the uterus toward the ovary. Here, if timing is right, sperm that have completed their journey encounter an egg recently released from the ovary. The egg, properly called a secondary oocyte at this stage, is not a solitary cell, but a complex surrounded by layers of protective cells and glycoproteins. The outermost layer is the corona radiata, consisting of several layers of granulosa cells that surrounded the oocyte in the follicle and were expelled with it during ovulation. These cells provide some nutrition and protection during the egg's brief viable period. Beneath the corona radiata lies the zona pellucida, a thick, transparent glycoprotein shell approximately 15 micrometers thick. The zona pellucida is not living tissue, but a secreted extracellular matrix produced earlier by the developing oocyte and surrounding granulosa cells. It consists primarily of three glycoproteins designated ZP1, ZP2, and ZP3, which form a cross-linked meshwork. The zona pellucida serves multiple crucial functions. It provides structural support, maintaining the egg's shape. It acts as a barrier that sperm must penetrate. It contains species-specific binding sites that ensure only human sperm can fertilize human eggs. And after fertilization, it prevents additional sperm from entering, avoiding polyspermy, which would be lethal. Inside the zona pellucida is the oocyte itself, a large cell approximately 120 micrometers in diameter, making it the largest cell in the human body and just visible to the naked eye as a tiny dot. The oocyte is arrested in metaphase 2 of meiosis, with chromosomes aligned on the metaphase plate, awaiting the signal to complete division. When capacitated sperm reach the egg, they encounter the corona radiata first. The cumulus cells of the corona radiata are held together by an extracellular matrix containing hyaluronic acid, a gel-like substance. Sperm must penetrate this matrix, which they accomplish through a combination of vigorous swimming and enzymatic digestion. The sperm surface contains hyaluronidase enzyme that breaks down hyaluronic acid, loosening the cumulus cells and creating a path. Multiple sperm typically reach the egg simultaneously, and many work together to disperse the corona radiata, though ultimately only one will fertilize the egg. This is not cooperation in any purposeful sense, but rather a byproduct of many sperm attacking the same barrier. After penetrating the corona radiata, sperm encounter the zona pellucida. Here, the process becomes more selective. The sperm plasma membrane contains proteins that recognize and bind to specific glycoproteins in the zona pellucida, particularly ZP3. This binding is species-specific. Human sperm recognize human ZP3 through complementary receptor molecules. When a sperm binds tightly to ZP3, it triggers the acrosome reaction, one of the most critical events in fertilization. The acrosome, that cap-like structure covering the anterior half of the sperm head, is essentially a large vesicle filled with hydrolytic enzymes. During the acrosome reaction, the outer acrosomal membrane fuses with the overlying sperm plasma membrane at multiple points. This fusion creates openings through which the acrosomal contents are released. The released enzymes include acrosin, a protease that digests proteins in the zona pellucida, and other hydrolases that break down the zona's structure. These enzymes create a path through the zona pellucida. The sperm, continuing its vigorous hyperactivated motility, thrusts forward through this path, burrowing through the zona like a drill through wood. The zona pellucida is approximately 15 micrometers thick, so the sperm must traverse a distance roughly 3 times its head length. This takes several minutes of continuous enzyme release and vigorous swimming. Only sperm that have undergone proper capacitation can execute the acrosome reaction and penetrate the zona. Uncapacitated sperm, even if they reach the egg, cannot fertilize it. Eventually, the sperm breaches the inner surface of the zona pellucida and enters the perivitelline space, a narrow fluid-filled gap between the zona pellucida and the egg's plasma membrane. The egg membrane, called the oolemma, is the final barrier. The sperm plasma membrane, now exposed by the acrosome reaction, contains proteins on its equatorial region that bind to receptors on the oolemma. When binding occurs, the two membranes fuse. This is a remarkable event. two separate cells become one. The fusion is mediated by specific proteins, including Izumo-1 on the sperm membrane and Juno on the egg membrane, named after Japanese and Roman deities associated with marriage. Once membrane fusion begins, it spreads rapidly. The sperm head, midpiece, and part of the tail are drawn into the egg cytoplasm. The sperm mitochondria and flagellum are usually internalized as well, though they will later degenerate. The human embryo inherits mitochondria only from the mother, not the father. The moment of sperm-egg fusion triggers a cascade of events within the egg called egg activation or cortical reaction. Within seconds, calcium ions flood into the egg cytoplasm. This calcium wave, propagating across the egg, initiates multiple processes. First and most immediately, cortical granules, vesicles positioned just beneath the ulema, fuse with the membrane and release their contents into the paravitelline space. The released contents include enzymes that modify the zona pellucida, cleaving ZP2 and hardening the entire structure. This zona reaction makes the zona pellucida impenetrable to additional sperm, preventing polyspermy. Polyspermy, the fertilization of an egg by more than one sperm, would result in a zygote with too many chromosomes, a condition incompatible with life. The zona reaction, occurring within minutes of the first sperm's entry, ensures that only one sperm's genetic material combines with the eggs. The calcium surge also triggers the egg to complete meiosis II. Remember, the egg has been arrested in metaphase 2 since shortly after ovulation. Now, stimulated by calcium, the metaphase spindle resumes activity. Sister chromatids separate, and the cell divides again, highly asymmetrically. One set of chromosomes remains in the large egg cell. The other set is extruded into a tiny second polar body, which sits in the paravitelline space and will eventually degenerate. The egg now contains a haploid set of 23 chromosomes, each as a single chromatid. Only now, after fertilization, does the egg technically become an ovum or mature egg, though the term egg is used more loosely. The sperm nucleus, now inside the egg cytoplasm, begins to swell and decondense. In the sperm, DNA is extremely tightly packed, with protamines replacing histones to compress the chromatin into the smallest possible volume. Now, the egg's machinery replaces protamines with histones, and the chromatin loosens. The swelling sperm nucleus is now called the male pronucleus. The egg's chromosomes, having completed meiosis II, reorganize into the female pronucleus. Both pronuclei migrate toward the center of the egg, drawn together by the egg's cytoskeleton. This migration takes several hours. As they move, both pronuclei replicate their DNA in preparation for the first mitotic division. When the pronuclei meet near the center, they do not actually fuse. Instead, their nuclear envelopes break down, and the chromosomes from both parents mingle in the egg cytoplasm. This is syngamy, the actual mixing of maternal and paternal genetic material. The chromosomes align on a mitotic spindle, just as in any cell preparing to divide. Now there are 46 chromosomes, 23 from the mother and 23 from the father, restoring the diploid number. The cell is now called a zygote, the first cell of the new individual. The zygote completes its first mitotic division, called cleavage, approximately 24 to 30 hours after fertilization. Unlike most cell divisions, where cells grow before dividing, cleavage divisions occur without growth. The zygote divides into 2 cells, then 4, then 8, and so on, with each cell called a blastomere. The total size remains roughly the same as the original egg, so the blastomeres become progressively smaller with each division. The timing of fertilization relative to ovulation is critical. The egg remains viable for only 12 to 24 hours after ovulation. If sperm do not reach and fertilize it within this window, the egg degenerates. Similarly, sperm require several hours in the female tract for capacitation. If intercourse occurs too close to ovulation, sperm may not be capacitated in time. Optimal conception occurs when intercourse happens 1 to 2 days before ovulation, giving sperm time to capacitate and be present when the egg arrives. Not all fertilization attempts succeed. Many eggs are not fertilized, despite sperm being present. The egg may have defects preventing sperm binding or penetration. The sperm may be unable to undergo the acrosome reaction. Binding may occur, but not membrane fusion. Even after fertilization, many embryos fail to develop properly due to chromosomal abnormalities or other defects. Chromosomal abnormalities become more common with maternal age. Eggs that have been arrested in meiosis for decades accumulate damage to the spindle apparatus and cellular machinery. When meiosis II completes at fertilization, chromosomes may not separate properly, resulting in aneuploid embryos with too many or too few chromosomes. Most aneuploid embryos fail to implant or miscarry shortly after implantation. Some, like those with trisomy 21, Down syndrome, develop to term but have developmental abnormalities. Occasionally, fertilization produces twins. Identical twins or monozygotic twins result when a single embryo splits into two separate embryos at an early stage, usually within the first week. Each develops into a genetically identical individual. Fraternal twins or dizygotic twins result when two eggs are released during ovulation and both are fertilized by different sperm. These twins are no more genetically similar than any siblings, simply sharing the womb and birthday. After fertilization, the conceptus, the term for the developing organism, begins a journey down the fallopian tube toward the uterus. This journey takes approximately 5 to 7 days, during which the embryo continues dividing and undergoing developmental changes that will prepare it for implantation in the uterine lining. But the moment of fertilization itself, when sperm and egg unite, represents the true beginning of a new human life, a unique genetic combination that has never existed before and will never exist again. After fertilization, the newly formed zygote begins a remarkable journey of development and travel. Over the next week, it will transform from a single cell into a complex multicellular structure while simultaneously traversing the fallopian tube and reaching the uterus, where it must implant into the endometrial lining to survive. This chapter of development occurs largely autonomously, with the embryo relying on nutrients stored in the egg and on secretions from the fallopian tube and uterus. The first major event after fertilization is cleavage, a series of rapid mitotic divisions. Approximately 30 hours after fertilization, the zygote completes its first division, producing two blastomeres, each containing 46 chromosomes in 23 pairs. These blastomeres are roughly half the size of the original zygote because there is no growth phase between divisions, unlike typical cell divisions where cells enlarge before dividing. The two-cell embryo continues dividing. Around 40 hours after fertilization, it becomes 4 cells. Around 50 to 60 hours, it reaches 8 cells. Each division is not perfectly synchronized, so briefly the embryo may have odd numbers of cells, such as 3 or 5, but the general pattern is doubling. Throughout these early divisions, The embryo remains enclosed within the zona pellucida, that glycoprotein shell that surrounded the egg. The zona pellucida, now hardened by the zona reaction, serves several protective functions. It maintains the embryo's structural integrity, preventing the blastomeres from dissociating. It provides a barrier against the maternal immune system, which might otherwise recognize the embryo as foreign tissue, and it prevents the embryo from prematurely implanting in the fallopian tube, which could lead to a dangerous ectopic pregnancy. By 72 to 96 hours after fertilization, the embryo typically reaches 16 to 32 cells. At this stage, it is called a morula, Latin for mulberry, because of its berry-like appearance. The morula is still roughly the same size as the original egg, approximately 120 micrometers in diameter, but now consists of tightly packed blastomeres rather than a single cell. During these early divisions, the embryo is traveling down the fallopian tube toward the uterus. This transport is not by the embryo's own locomotion, but is facilitated by muscular contractions in the tube wall and by the beating of cilia lining the tube's epithelium. These cilia beat in waves, creating currents in the fluid surrounding the embryo, gently propelling it toward the uterus. The journey is relatively slow, taking approximately 3 to 5 days to traverse the 10 to 12 centimeter length of the tube. This timing is crucial. The embryo must not reach the uterus too early before the endometrium is ready to receive it, nor too late when the endometrium may already be regressing. The embryo's developmental stage must synchronize with the uterine state. As the morula travels, fluid begins to accumulate within it. Gap junctions between blastomeres allow ions and small molecules to pass, but tight junctions form between some cells, creating a barrier. Sodium ions are actively transported into the interior of the morula, and water follows osmotically. A fluid-filled cavity, called the blastocoel, forms in the center. As the blastocoel expands, the morula transforms into a blastocyst. This transformation occurs around day 4 to 5 after fertilization, typically as the embryo enters the uterus or shortly thereafter. The blastocyst is a hollow sphere of cells with a fluid-filled cavity. The cells of the blastocyst differentiate into two distinct populations. The trophoblast is a single layer of cells forming the outer wall of the sphere surrounding the blastocoel. These cells will not contribute to the embryo itself, but will form the placenta and other supporting structures. The inner cell mass, also called the embryoblast, is a cluster of cells positioned at one pole of the blastocyst, attached to the inner surface of the trophoblast. These cells will form the actual embryo, all the tissues and organs of the developing individual. The entire complex of trophoblast, blastocoel, and inner cell mass is still enclosed within the zona pellucida. The blastocyst stage represents the first differentiation in development. Up to this point, all blastomeres were essentially equivalent. Now, cells have taken on different fates. The mechanisms determining which cells become trophoblast and which become inner cell mass involve complex signaling and gene expression patterns. Cells on the outside, exposed to the environment, receive different signals than cells in the interior, and these position-dependent signals influence cell fate. Once in the uterus, the blastocyst floats freely for 1 to 2 days, bathed in uterine secretions. During this time, it continues to expand, and the blastocoel enlarges. The embryo is now relying entirely on nutrients from uterine secretions, as the maternal resources stored in the original egg are largely depleted. Around day 5 to 6 after fertilization, the blastocyst undergoes hatching, escaping from the zona pellucida. Enzymatic digestion weakens the zona, and the expanding blastocyst exerts pressure from within. Eventually, the zona splits, and the blastocyst squeezes through the opening, emerging from the shell that has enclosed it since it was an egg. Hatching is essential for implantation. The zona pellucida prevents direct contact with the uterine lining. Once free, the trophoblast cells can interact with the endometrium. The timing of hatching is critical. It must occur when the endometrium is receptive, during the narrow window of implantation, approximately days 20 to 24 of the menstrual cycle. The endometrium at this stage is in the secretory phase, transformed by progesterone from the corpus luteum. It is thick, richly vascularized, and its glands are secreting glycogen-rich fluid. The stromal cells have undergone decidualization, swelling with glycogen and lipids. This is the optimal state for receiving an embryo. Implantation begins around day 6 to 7 after fertilization, corresponding to days 20 to 21 of a typical menstrual cycle. The process starts with apposition, where the blastocyst orients itself with the inner cell mass side toward the endometrium and contacts the uterine surface. The trophoblast cells in contact with the endometrium are called the polar trophoblast, as they are at the pole containing the inner cell mass. The polar trophoblast cells adhere to the endometrial epithelium through interactions between adhesion molecules on the trophoblast surface and the endometrial cells. This initial attachment is reversible, and the blastocyst may detach and reattach elsewhere if conditions are not optimal. Once firmly attached, the trophoblast begins to invade. The cells undergo rapid proliferation and differentiation. Two distinct layers emerge. The inner layer remains cellular with distinct cell boundaries and is called the cytotrophoblast. The outer layer undergoes a remarkable transformation. The cell membranes break down, and the cells fuse into a multinucleated mass without distinct cell boundaries, forming the syncytiotrophoblast. The syncytiotrophoblast is highly invasive. It secretes enzymes that digest the endometrial epithelium and underlying connective tissue. These proteolytic enzymes break down the extracellular matrix, creating space for the blastocyst to penetrate. The syncytiotrophoblast extends finger-like projections that burrow into the endometrium. As the syncytiotrophoblast invades, it disrupts maternal blood vessels in the endometrium. Small pools of maternal blood form around the invading trophoblast. The trophoblast extensions, now forming the beginnings of chorionic villi, are bathed in this maternal blood, establishing primitive exchange of nutrients and gases. The embryo, though only a tiny collection of cells, requires oxygen and nutrients, and the mother's blood provides them. The invasion continues until the entire blastocyst is embedded within the endometrium. This typically occurs by day 10 to 12 after fertilization. The endometrial surface epithelium regenerates over the site of implantation, completely enclosing the embryo within the uterine wall. From the outside, there may be no visible sign that an embryo has implanted, though some women experience slight spotting, called implantation bleeding, caused by disruption of small blood vessels. Implantation is not always successful. The embryo may be developmentally abnormal and fail to produce the signals necessary for implantation. The endometrium may not be adequately prepared if hormonal timing is off. The embryo may implant in an abnormal location, such as remaining in the fallopian tube, causing an ectopic pregnancy, which is dangerous and non-viable. Even normal embryos in receptive endometrium may fail to implant for reasons not entirely understood. It is estimated that 30 to 50% of fertilized eggs fail to implant successfully. Once implanted, the syncytia trophoblast begins secreting human chorionic gonadotropin, or HCG. This hormone is structurally similar to LH and binds to LH receptors on the corpus luteum. HCG rescues the corpus luteum from its programmed degeneration, maintaining its production of progesterone and estrogen. This hormonal rescue is critical. Progesterone maintains the endometrium, preventing menstruation. Without HCG, the corpus luteum would degenerate around day 28, progesterone would plummet, and the endometrium would shed, flushing out the embryo. HCG prevents this, signaling the mother's body that pregnancy has occurred and the endometrium must be maintained. HCG levels rise rapidly in early pregnancy, doubling approximately every 48 hours. This rapid rise is the basis for pregnancy tests, which detect HCG in urine or blood. A positive pregnancy test indicates that implantation has occurred and the embryo is producing HCG. While implantation proceeds, the inner cell mass continues developing. It differentiates into two layers. The epiblast, closer to the blastocoel, will form the embryo proper. The hypoblast, adjacent to the blastocoel cavity, will form the yolk sac, a supporting structure. Between the epiblast and trophoblast, a new cavity forms, the amniotic cavity, which will eventually surround the developing embryo. By the end of the second week after fertilization, the conceptus has transformed from a simple blastocyst into a complex structure embedded in the uterine wall. The trophoblast is actively invading and establishing connections with maternal blood vessels. The inner cell mass has differentiated into epiblast and hypoblast. The foundations are laid for the placenta, amniotic sac, and embryo itself. Week 3 marks the beginning of gastrulation, the process by which the embryo transforms from 2 layers into 3 germ layers: ectoderm, mesoderm, and endoderm. These germ layers will give rise to all the tissues and organs of the body. Ectoderm will form the nervous system, skin, and sensory organs. Mesoderm will form muscles, bones, cardiovascular system, and reproductive organs. Endoderm will form the lining of the digestive and respiratory systems and associated organs. Gastrulation is initiated by the formation of the primitive streak, a groove that appears on the surface of the epiblast. Cells migrate through the primitive streak, moving inward and spreading out between the epiblast and hypoblast, forming the mesoderm. Some cells displace the hypoblast, forming the definitive endoderm. The remaining epiblast becomes ectoderm. This 3-layered structure, called the trilaminar embryonic disc, is the foundation for all subsequent development. During week 3, the embryo also begins to establish its body plan. The primitive streak defines the anterior-posterior axis, head to tail. The notochord, a rod-like structure forming from mesoderm, defines the midline and will induce the formation of the neural tube, the precursor to the brain and spinal cord. By the end of week 3, the embryo is approximately 2 millimeters long and contains perhaps a few thousand cells organized into 3 germ layers. It is still tiny, barely visible to the naked eye, but the fundamental architecture of the body is emerging. The cardiovascular system begins forming, with the heart starting to beat around day 21 to 22, even though the embryo is only 3 weeks old. Throughout this early development, the embryo is critically vulnerable. Environmental factors, maternal health, nutritional status, and exposure to toxins can profoundly affect development. Many pregnancies end in early miscarriage, often before the woman even knows she is pregnant. It is estimated that 50 to 75% of fertilized eggs do not result in live birth, with most losses occurring in the first few weeks. The causes of early pregnancy loss are varied. Chromosomal abnormalities are the most common cause, accounting for more than half of early miscarriages. Hormonal insufficiencies, particularly inadequate progesterone production, can prevent the endometrium from sustaining the embryo. Immune factors may cause rejection of the embryo. Uterine abnormalities can prevent proper implantation. Many losses have no identifiable cause. Despite these challenges, the resilience of early development is remarkable. From a single fertilized egg, a complex multicellular organism emerges, differentiates into specialized cell types, and begins forming organs, all while embedded in the uterine wall and connected to maternal circulation. The precision required, the timing, the coordination of cellular processes, all occurring autonomously according to genetic programming, represents one of biology's most extraordinary achievements. As the embryo completes implantation and begins forming its basic body structure, the placenta also begins developing. This remarkable temporary organ will be the embryo's lifeline throughout pregnancy, mediating all exchanges between mother and fetus and producing hormones that maintain pregnancy. The placenta's development and function are our next focus. The placenta is a remarkable temporary organ, existing for only 9 months, yet serving functions critical to the developing fetus. It acts as lungs, providing oxygen and removing carbon dioxide. It serves as a digestive system, delivering nutrients and removing metabolic wastes. It functions as an endocrine gland, producing hormones that maintain pregnancy and regulate maternal physiology. And it acts as a selective barrier, protecting the fetus from many harmful substances while allowing beneficial materials through. The placenta represents the interface between two individuals, mother and fetus, maintaining their physiological separation while enabling the exchange necessary for fetal survival. Placental development begins during implantation, when the trophoblast invades the endometrium and contacts maternal blood. The trophoblast, remember, differentiates into 2 layers, the inner cytotrophoblast and the outer syncytiotrophoblast. As the syncytiotrophoblast invades deeper into the endometrium, it erodes maternal blood vessels, creating pools of maternal blood called lacunae. These blood-filled spaces surround extensions of trophoblast tissue, establishing the basic architecture for exchange. During the 2nd week after fertilization, the trophoblast develops projections called primary chorionic villi. These are initially just finger-like extensions of cytotrophoblast, covered by syncytiotrophoblast, protruding into the lacunae. Soon, mesoderm from the embryo migrates into the villi, forming a connective tissue core. These are secondary villi. By the 3rd week, blood vessels develop within the mesodermal core, forming tertiary villi. These blood vessels connect to the embryo's developing cardiovascular system. The mature chorionic villus, the functional unit of the placenta, has a characteristic structure. At its core are fetal blood vessels, arterioles and capillaries, carrying deoxygenated blood from the fetus. Surrounding the vessels is connective tissue derived from mesoderm. Outside this is a layer of cytotrophoblast cells, though this layer becomes discontinuous later in pregnancy. Encoding everything is the syncytiotrophoblast, a multinucleated layer without distinct cell boundaries in direct contact with maternal blood. The syncytiotrophoblast is the primary barrier between maternal and fetal circulations. Despite its name suggesting a barrier, it is actually quite permeable, designed to facilitate exchange. Its surface area is enormous, approximately 11 to 13 square meters at term, roughly the size of a tennis court, providing vast surface for diffusion. Between the chorionic villi and maternal tissues is the intervillous space filled with maternal blood. Maternal spiral arteries, specialized blood vessels in the endometrium, open directly into this space, spurting blood that bathes the villi. The blood flows slowly through the intervillous space, allowing time for exchange, then drains into maternal veins that return it to circulation. Maternal blood flow through the placenta at term is approximately 500 to 750 milliliters per minute, representing about 10 to 15% of maternal cardiac output. Fetal blood flows through the capillaries within the villi. Deoxygenated blood arrives through two umbilical arteries, spreads through the capillary networks in millions of villi, picks up oxygen and nutrients while releasing carbon dioxide and wastes, then returns to the fetus through a single umbilical vein. Fetal blood flow through the placenta at term is approximately 400 milliliters per minute. Despite the intimate contact, maternal and fetal blood do not mix. They are separated by the placental barrier, consisting of the syncytiotrophoblast, cytotrophoblast when present, connective tissue, and fetal capillary endothelium. This barrier is analogous to the blood-air barrier in the lungs, separating two circulations while allowing exchange. Exchange across the placenta occurs through multiple mechanisms. Simple diffusion is the primary mechanism for oxygen, carbon dioxide, and lipid-soluble substances. These molecules move down their concentration gradients. Oxygen diffuses from maternal blood, where its partial pressure is higher, into fetal blood. Carbon dioxide diffuses in the opposite direction. The thin syncytiotrophoblast and capillary endothelium present minimal resistance to diffusion of these small, lipid-soluble gases. Fetal hemoglobin has higher oxygen affinity than adult hemoglobin, a critical adaptation. At the oxygen partial pressures present in the placenta, fetal hemoglobin binds oxygen more avidly than maternal. Hemoglobin releases it. This ensures efficient oxygen transfer from mother to fetus, despite the relatively low oxygen environment of the placenta compared to the lungs. Water-soluble substances like glucose, amino acids, and ions require facilitated diffusion or active transport. The syncytiotrophoblast contains numerous transporter proteins embedded in its membranes. Glucose crosses via GLUT transporters, moving down its concentration gradient, but requiring specific transport proteins. Maternal blood glucose levels are typically higher than fetal levels, ensuring continuous glucose supply to the fetus. Amino acids are actively transported against their concentration gradients using ATP-dependent transporters. This ensures the fetus receives adequate protein building blocks, even if maternal amino acid levels are modest. Similarly, calcium, iron, and vitamins are actively transported, often accumulating in fetal blood at concentrations higher than maternal blood. Larger molecules like proteins generally cannot cross the placental barrier. However, certain proteins are transferred through receptor-mediated endocytosis. Immunoglobulin G, or IgG, maternal antibodies that provide passive immunity to the fetus, are transported via FcRn receptors. The syncytia trophoblast binds IgG from maternal blood, internalizes it in vesicles, and releases it on the fetal side. This transfer provides the newborn with temporary immunity to diseases the mother has encountered, lasting several months until the infant's own immune system matures. The placenta is selective, but not perfectly so. Some substances that should be excluded can cross. Alcohol is lipid soluble and crosses easily, which is why maternal alcohol consumption can damage the developing fetus. Many drugs, both therapeutic and recreational, cross the placenta. Nicotine from cigarettes crosses readily. Some pathogens can cross, including Treponema pallidum causing syphilis, Toxoplasma gondii, cytomegalovirus, and Zika virus. HIV can cross, particularly during labor. The placenta provides substantial, but not absolute protection. The placenta is also an endocrine organ, producing hormones essential for maintaining pregnancy and preparing the mother's body for childbirth and lactation. Human chorionic gonadotropin, HCG, is the first hormone produced, secreted by the syncytia trophoblast beginning at implantation. As discussed, HCG maintains the corpus luteum, ensuring continued progesterone and estrogen production during early pregnancy. HCG levels rise rapidly, doubling every 48 hours, peaking around 8 to 11 weeks of gestation. After this peak, levels decline to a plateau that is maintained throughout pregnancy. The initial high levels are thought to contribute to nausea and vomiting of early pregnancy, commonly called morning sickness, though it can occur at any time of day. By week 10 to 12 of pregnancy, the placenta itself begins producing progesterone and estrogen, gradually taking over from the corpus luteum. Placental progesterone production increases steadily throughout pregnancy, reaching levels far higher than during the menstrual cycle. Progesterone maintains the endometrium, suppresses uterine contractions, and suppresses maternal immune responses against the fetus. The placenta produces multiple estrogens, primarily estriol. Estrogen stimulates growth of the uterus to accommodate the growing fetus, increases blood flow to the uterus, and promotes breast development in preparation for lactation. Estrogen levels rise steadily throughout pregnancy, reaching peaks far exceeding non-pregnant levels. The placenta also produces human placental lactogen, HPL, also called human chorionic somatomamotropin. This hormone has effects similar to growth hormone and prolactin. It promotes maternal protein synthesis and mobilization of maternal fat stores, ensuring adequate nutrients for the fetus. It also induces insulin resistance in the mother, raising maternal blood glucose levels and ensuring continuous glucose availability for the fetus. Other placental hormones include relaxin, which softens the cervix and loosens pelvic ligaments in preparation for childbirth, and corticotropin-releasing hormone, which increases late in pregnancy and may contribute to initiating labor. The umbilical cord connects the fetus to the placenta. It contains 2 umbilical arteries and 1 umbilical vein embedded in a gelatinous connective tissue called Wharton's jelly. Despite their names, the umbilical arteries carry deoxygenated blood from the fetus to the placenta, and the umbilical vein carries oxygenated blood from placenta to fetus. This naming reflects the direction of flow relative to the fetus rather than oxygen content. The umbilical cord at term is typically 50 to 60 centimeters long and about 1.5 to 2 centimeters in diameter. It is remarkably strong and flexible, capable of withstanding compression and torsion as the fetus moves. Wharton's jelly provides cushioning and prevents the blood vessels from kinking or collapsing. The fetal circulation is substantially different from postnatal circulation because the fetal lungs are non functional, filled with fluid rather than air. Blood must bypass them. The fetus has specialized vascular shunts that redirect blood flow. The ductus venosus allows some oxygenated blood returning from the placenta via the umbilical vein to bypass the liver and flow directly to the inferior vena cava and heart. The foramen ovale is an opening between the right and left atria, allowing oxygenated blood entering the right atrium to flow directly to the left atrium, bypassing the right ventricle and lungs. The ductus arteriosus connects the pulmonary artery to the aorta, shunting blood from the right ventricle away from the lungs and into systemic circulation. These shunts close shortly after birth when the infant takes its first breaths and the lungs expand. The change in pressure gradients causes the foramen ovale to close. The ductus arteriosus constricts in response to increased oxygen levels. The ductus venosus closes when the umbilical cord is clamped. These closures redirect blood through the lungs, establishing the adult circulatory pattern. The placenta grows throughout pregnancy, though its growth rate slows relative to the fetus in the third trimester. At term, the placenta is approximately 20 to 22 centimeters in diameter, 2 to 3 centimeters thick, and weighs about 500 to 600 grams, roughly 1/6 of the baby's weight. Placental efficiency increases as pregnancy progresses. Early in pregnancy, the placental barrier is thicker, consisting of more cell layers. Later, the cytotrophoblast layer becomes discontinuous and the syncytiotrophoblast thins, reducing the diffusion distance. This compensates for the increasing metabolic demands of the growing fetus. Placental function can be compromised by various conditions. Placental insufficiency, inadequate placental function, can result from maternal hypertension, diabetes, smoking, or placental abnormalities. Insufficient exchange leads to fetal growth restriction, where the fetus does not grow at expected rates. Placenta previa occurs when the placenta implants low in the uterus, partially or completely covering the cervix. This can cause bleeding and necessitates delivery by cesarean section. Placental abruption is premature separation of the placenta from the uterine wall, a medical emergency that deprives the fetus of oxygen. Preeclampsia is a pregnancy complication characterized by maternal hypertension and protein in the urine, thought to originate from abnormal placental development and function. In severe cases, it can progress to eclampsia, involving seizures, and HELP syndrome, involving hemolysis, elevated liver enzymes, and low platelets. These conditions threaten both mother and fetus. Despite these potential complications, the placenta usually functions remarkably well, supporting fetal growth from a few grams to 3 to 4 kilograms over 9 months. The placenta is unique among human organs in its temporary existence and its formation from tissue derived from both mother, the decidualized endometrium, and fetus, the chorionic villi. At birth, the placenta is expelled shortly after the infant, its mission complete. This temporary organ that sustained life for 9 months is discarded, having served its purpose. With the placenta formed and functioning, supporting the developing fetus with oxygen, nutrients, and hormones, we can now turn our attention to the broader changes occurring in the mother's body as pregnancy progresses, the profound adaptations that allow a woman to sustain another life within her own. Pregnancy is a remarkable state of physiological upheaval. For roughly 40 weeks, or more precisely, 280 days from the last menstrual period, the maternal body undergoes a series of profound adaptations to accommodate a growing human being. These changes are not merely cosmetic or superficial. They represent a fundamental restructuring of nearly every major organ system, a biological commitment to supporting another life at considerable metabolic expense. The cardiovascular system, predictably, bears much of this burden. Blood volume increases substantially by approximately 40 to 50% above non-pregnant levels. This expansion begins early, around the 6th week of gestation, and reaches its peak by the 32nd to 34th week. The extra volume is necessary to supply the placenta, support the enlarging uterus, and prepare for the inevitable blood loss during delivery. Plasma volume increases more than red blood cell mass, resulting in what's called physiological anemia of pregnancy. The hemoglobin concentration drops slightly, not because red blood cells are being destroyed, but because they're diluted in a larger pool of plasma. This is normal, expected, and generally well tolerated. Cardiac output, the amount of blood pumped by the heart per minute, increases by 30 to 50%. This rise is achieved through a combination of increased heart rate, typically 10 to 15 beats per minute faster than baseline, and increased stroke volume, the amount of blood ejected with each heartbeat. The heart literally works harder, beating faster, and pushing more blood with each contraction. By the 3rd trimester, a pregnant woman's heart is pumping an additional 1.5 liters of blood every minute compared to her non-pregnant state. Blood pressure, interestingly, follows a somewhat paradoxical pattern. Despite the increased blood volume and cardiac output, blood pressure typically decreases during the 2nd trimester. This occurs because progesterone causes systemic vasodilation, relaxing the smooth muscle in arterial walls and reducing vascular resistance. The result is a drop in both systolic and diastolic pressures, often by 5 to 10 millimeters of mercury. By the 3rd trimester, blood pressure gradually returns to pre-pregnancy levels. This U-shaped curve in blood pressure is one of pregnancy's curious adaptations, a temporary softening of the vascular system to accommodate increased flow. The enlarging uterus creates its own set of mechanical challenges for circulation. As it grows, it can compress the inferior vena cava, the large vein returning blood from the lower body to the heart, particularly when a woman lies flat on her back. This compression can reduce venous return, decrease cardiac output, and cause dizziness or even fainting, a condition known as supine hypotensive syndrome. This explains the obstetric recommendation for pregnant women to sleep on their left side during the later months, a position that shifts the uterus away from the vena cava and preserves blood flow. The respiratory system undergoes its own adaptations. The diaphragm, the primary muscle of breathing, is pushed upward by as much as 4 centimeters as the uterus expands into the abdominal cavity. One might expect this displacement to impair breathing, and indeed, many pregnant women report a sensation of breathlessness, particularly in the 3rd trimester. Yet, paradoxically, lung function actually improves in some respects. Tidal volume, the amount of air inhaled with each breath, increases by 30 to 40%. This means that with each breath, a pregnant woman draws in more air than she did before pregnancy, compensating for the reduced space available for lung expansion. The increased tidal volume is driven by progesterone, which acts as a respiratory stimulant. It makes the respiratory center in the brainstem more sensitive to carbon dioxide, prompting deeper, more frequent breaths. The result is a state of mild hyperventilation, with lower levels of carbon dioxide in the blood than in the non-pregnant state. This isn't harmful. In fact, it facilitates the transfer of carbon dioxide from fetal blood to maternal blood across the placenta since gases move down concentration gradients. The sensation of breathlessness that many pregnant women experience, dyspnea, is not due to inadequate oxygen supply. Oxygen levels remain normal or even slightly elevated. Rather, it seems to be related to the increased work of breathing and the awareness of one's own respiration, a heightened consciousness of the act itself. It's uncomfortable but not dangerous, one of pregnancy's many minor inconveniences. The renal system, responsible for filtering blood and producing urine, also adjusts to pregnancy's demands. The kidneys enlarge slightly, and the glomerular filtration rate, the speed at which blood is filtered, increases by 40 to 50%. This means the kidneys are working harder, processing more blood per minute than usual. As a consequence, substances that are normally reabsorbed, like glucose and amino acids, may occasionally appear in the urine simply because the kidneys can't keep up with the increased load. Small amounts of glucose in the urine, glycosuria, can be normal in pregnancy, though it warrants monitoring to rule out gestational diabetes. The increased kidney function also means that waste products like urea and creatinine are cleared more efficiently, resulting in lower blood concentrations of these substances. This is generally beneficial, though it does make interpreting standard lab values more complicated since what's normal for a pregnant woman differs from what's normal for everyone else. Frequent urination, one of pregnancy's hallmark symptoms, has two primary causes. Early in pregnancy, it's largely due to hormonal changes and increased blood flow to the kidneys, which produce more urine. Later, as the uterus enlarges and presses against the bladder's capacity is reduced, creating a persistent sense of fullness and the need to urinate more often. It's a mechanical problem compounded by a physiological one, and there's little to be done about it except to make frequent trips to the bathroom. The gastrointestinal system experiences significant and often unpleasant changes. Nausea and vomiting, colloquially known as morning sickness, though it can occur at any time of day, affect a majority of pregnant women, particularly in the first trimester. The exact cause remains somewhat mysterious, though rising levels of human chorionic gonadotropin, HCG, and estrogen are likely culprits. For most women, the symptoms resolve by the second trimester, though a small percentage experience persistent nausea throughout pregnancy, and an even smaller fraction develop hyperemesis gravidarum, a severe form requiring medical intervention. Heartburn, or gastroesophageal reflux, becomes increasingly common as pregnancy progresses. Progesterone relaxes the lower esophageal sphincter, the muscular valve between the esophagus and stomach, allowing stomach acid to creep upward. The expanding uterus also increases intra-abdominal pressure, physically pushing stomach contents toward the esophagus. The result is a burning sensation in the chest, particularly after eating or when lying down. Constipation is another frequent complaint. Progesterone slows gastrointestinal motility, meaning food moves more slowly through the intestines. This allows more time for water absorption, resulting in harder, drier stools. The pressure of the uterus on the intestines and rectum doesn't help matters. Iron supplements, commonly prescribed during pregnancy, can exacerbate the problem. The solution typically involves increased fiber intake, adequate hydration, and sometimes a stool softener. The musculoskeletal system adapts to the shifting center of gravity caused by the enlarging uterus. As the abdomen protrudes forward, the lumbar spine compensates by increasing its curve, a posture called lordosis. This helps maintain balance but places additional strain on the lower back, contributing to the back pain that plagues many pregnant women, particularly in the third trimester. The hormone relaxin, produced by the corpus luteum and placenta, loosens ligaments and connective tissue throughout the body in preparation for childbirth. This increased laxity is particularly important in the pelvis, where it allows the pelvic bones to shift slightly during delivery, accommodating the passage of the baby's head. However, the systemic effects of relaxin can destabilize joints elsewhere, contributing to discomfort in the hips, knees, and lower back. Weight gain during pregnancy is variable, but typically ranges from 25 to 35 pounds for women of normal pre-pregnancy weight. This isn't simply the weight of the baby. By term, the fetus accounts for only about 7 to 8 pounds. The placenta adds another pound or so, and the amniotic fluid contributes roughly 2 pounds. The enlarged uterus, increased blood volume, expanded breast tissue, and stored maternal fat make up the remainder. This stored fat, often frustrating to women concerned about their appearance, serves an important purpose. It provides an energy reserve for lactation after birth. Metabolically, pregnancy is a state of insulin resistance, particularly in the second and third trimesters. This means that the body's cells become less responsive to insulin, requiring more of the hormone to maintain normal blood glucose levels. The placenta produces hormones, including human placental lactogen, that oppose insulin's action, ensuring that glucose remains available in the bloodstream for the fetus, which depends entirely on maternal glucose for energy. For most women, the pancreas compensates by producing more insulin, and blood sugar remains normal. However, some women cannot produce enough insulin to overcome the resistance, resulting in gestational diabetes, a temporary form of diabetes that typically resolves after delivery but increases the risk of developing type 2 diabetes later in life. Nutritional needs increase during pregnancy, though not as dramatically as popular wisdom suggests. The old adage about eating for two is misleading. Caloric needs increase by only about 300 to 500 calories per day, a modest amount. More important than quantity is quality. Protein requirements increase to support fetal growth, particularly in the second and third trimesters. Folic acid is critical in the early weeks to prevent neural tube defects, which is why supplementation is recommended even before pregnancy is confirmed. Iron is needed to support the expanded red blood cell mass, and calcium is essential for fetal bone development. The developing fetus follows a predictable trajectory, though with considerable individual variation. Pregnancy is typically divided into 3 trimesters, each roughly 13 weeks long, and each characterized by distinct developmental milestones. The first trimester, weeks 1 through 13, is the period of organogenesis, during which all major organ systems form. By the end of the 8th week, the embryo, now called a fetus, has recognizable human features, a head, trunk, and limbs, though it measures only about an inch in length. The heart, which began beating around week 5, is fully formed with 4 chambers. The brain undergoes rapid development, and the foundation for all other organs is established. This is the most critical period for development, the time when the fetus is most vulnerable to teratogens, substances that can cause birth defects. Alcohol, certain medications, infections, and environmental toxins can all interfere with organogenesis, sometimes with devastating consequences. By the end of the first trimester, the fetus is about 3 inches long and weighs less than an ounce. It's moving, though these movements are not yet perceptible to the mother. The external genitalia have developed, making it possible to determine sex via ultrasound, though this is often deferred until the second trimester when visualization is clearer. The second trimester, weeks 14 through 27, is often considered the most comfortable period of pregnancy. Morning sickness typically subsides, energy levels improve, and the abdomen, while growing, is not yet large enough to cause significant discomfort. This is the period of rapid growth and maturation. The fetus grows from about 3 inches to roughly 14 inches in length and increases in weight from less than an ounce to about 2 pounds. Around week 20, most women begin to feel fetal movements, a sensation called quickening. These early movements are often described as flutters or bubbles, easily mistaken for gas. As the fetus grows stronger, the movements become unmistakable kicks, jabs, and rolls. By the end of the second trimester, the fetus has developed a sleep-wake cycle, periods of activity alternating with periods of rest, often maddeningly misaligned with the mother's own schedule. The lungs, though structurally formed, are not yet functional. The fetus practices breathing movements, drawing amniotic fluid in and out of the lungs, but gas exchange still occurs entirely through the placenta. Surfactant, the substance that prevents the alveoli from collapsing, begins to be produced late in the second trimester, though not yet in sufficient quantities to support independent breathing. The third trimester, weeks 28 through 40, is the period of final growth and preparation for birth. The fetus gains weight rapidly, accumulating fat stores that will help with temperature regulation after birth. By 28 weeks, the fetus has reached viability, the point at which survival outside the womb is possible with medical assistance. Viability doesn't mean easy survival. Babies born this early face significant challenges, including respiratory distress, feeding difficulties, and a host of potential complications. However, with advances in neonatal care, survival rates for babies born at 28 weeks and beyond have improved dramatically. By 32 weeks, the lungs are producing adequate surfactant, and the risk of respiratory complications decreases substantially with each passing week. By 37 weeks, a pregnancy is considered full term, and the baby is developmentally ready for life outside the womb. Babies born between 37 and 42 weeks are classified as term births, the goal for any pregnancy. During the final weeks, the fetus typically settles into a head-down position called vertex presentation in preparation for birth. The head, being the largest part of the body, engages in the pelvis, a process called lightening, which may relieve pressure on the diaphragm and improve breathing for the mother, but increases pressure on the bladder. Throughout pregnancy, the fetus is monitored indirectly through the mother. Weight gain, fundal height, the measurement from the pubic bone to the top of the uterus, and fetal heart rate all provide information about fetal well-being. Ultrasound offers visual confirmation of growth and development, detecting potential problems like structural abnormalities or issues with the placenta or amniotic fluid volume. Pregnancy is not a passive state for the fetus. It's actively preparing for birth and the transition to extrauterine life. The digestive system practices by swallowing amniotic fluid. The kidneys produce urine, which is released into the amniotic sac, contributing to its volume. The skin, initially translucent, thickens and loses most of the fine hair, lanugo, that covered it earlier. The bones harden, though the skull remains flexible, with soft spots called fontanelles that allow the head to compress slightly during passage through the birth canal. As the due date approaches, hormonal changes signal that labor is near. The exact trigger for labor remains incompletely understood, but it appears to involve a complex interaction between fetal and maternal signals, a coordinated transition from the quiet gestation of pregnancy to the dramatic process of birth. Labor is the culmination of pregnancy, the physiological process by which the fetus, placenta, and membranes are expelled from the uterus. It's a dramatic transition, both for mother and child, and it unfolds in three distinct stages. The first stage involves the dilation of the cervix, the second, the delivery of the baby, and the third, the expulsion of the placenta. Each stage has its own characteristics, its own challenges, and its own timeline, though considerable variation exists from one birth to another. The onset of labor is typically preceded by signs that the body is preparing for birth. In the days or weeks before true labor begins, many women experience Braxton Hicks contractions, irregular, often painless tightenings of the uterus that have been occurring throughout pregnancy but become more noticeable in the final weeks. These are practice contractions of sorts, a rehearsal rather than the main event. True labor contractions are different. They're regular, increasing in frequency and intensity, and they don't stop with rest or position changes. The exact trigger for labor remains one of obstetrics' enduring mysteries, though several pieces of the puzzle have been identified. Late in pregnancy, the fetus produces cortisol, a stress hormone that signals maturity. This fetal cortisol stimulates the placenta to increase production of estrogen while decreasing progesterone production. The resulting shift in the estrogen to progesterone ratio is critical. Throughout pregnancy, progesterone has maintained uterine quiescence, suppressing contractions. As its dominance wanes and estrogen rises, the uterus becomes more excitable, more responsive to stimuli. Estrogen increases the number of oxytocin receptors on the smooth muscle cells of the uterus, priming them to respond to oxytocin, a hormone produced by the posterior pituitary gland. Estrogen also stimulates the production of prostaglandins, lipid compounds that soften and thin the cervix, a process called ripening, and promote uterine contractions. Prostaglandins are so effective at inducing labor that synthetic versions are sometimes used medically to induce or augment labor when necessary. Oxytocin plays a central role in the initiation and progression of labor. As labor begins, oxytocin is released in pulses, stimulating rhythmic uterine contractions. These contractions push the baby's head against the cervix, stretching it and sending signals back to the brain via sensory nerves. This stretch triggers the release of more oxytocin, which intensifies the contractions, which in turn increases cervical stretch, prompting even more oxytocin. This is a positive feedback loop, a relatively rare phenomenon in human physiology. Most regulatory systems operate on negative feedback, dampening responses to maintain equilibrium. Labor, however, requires escalation, a building intensity that culminates in delivery. The first stage of labor, cervical dilation, is typically the longest, lasting an average of 12 to 16 hours for first-time mothers and somewhat shorter, 8 to 10 hours, for women who have given birth before. This stage is further divided into two phases, the latent phase and the active phase. During the latent phase, contractions are relatively mild and irregular, and the cervix dilates slowly from 0 to about 3 or 4 centimeters. This phase can last for hours or even days, and it's often spent at home, where the woman can move around, rest, and conserve energy for the work ahead. Contractions during this phase are typically 5 to 20 minutes apart and last 30 to 60 seconds. They're uncomfortable but usually manageable with breathing techniques, position changes, and distractions. The active phase begins when the cervix reaches about 4 centimeters of dilation and continues until full dilation at 10 centimeters. This phase is more intense. Contractions become stronger, longer, and closer together, typically occurring every 3 to 5 minutes and lasting 45 to 90 seconds. The pain intensifies as the uterine muscle contracts with increasing force. This is when many women seek pain relief, whether through epidural anesthesia, which blocks nerve transmission from the lower body, or other methods. The cervix, normally a firm, elongated structure about 3 to 4 centimeters in length, undergoes remarkable changes during labor. It effaces, shortening and thinning until it becomes part of the lower uterine segment, and it dilates, opening from a pinpoint to a diameter of 10 centimeters, wide enough to allow the baby's head to pass through. This transformation is the result of both the softening effects of prostaglandins and the mechanical pressure of contractions and the descending fetal head. As the cervix dilates, the baby descends through the pelvis, navigating a passage that's both bony and soft. The pelvis is not a simple cylindrical tube. It has curves, narrowings, and protrusions. The baby must twist and turn, adjusting its position to fit through the narrowest points. This process is called descent, and it occurs gradually throughout the first stage and into the second stage of labor. The baby's head is the largest part of its body, and fortunately, it's somewhat compressible. The skull bones are not yet fused, and the spaces between them, the sutures, allow for a degree of overlapping called molding. This molding can give the baby's head an elongated, cone-like shape immediately after birth, a temporary deformation that resolves within days as the bones return to their normal positions. The second stage of labor begins when the cervix is fully dilated at 10 centimeters and ends with the delivery of the baby. This stage is typically shorter than the first, lasting an average of 1 to 2 hours for first-time mothers and 20 minutes to an hour for multiparous women, those who have given birth before. However, these are averages, and the range of normal is broad. During the second stage, the nature of contractions changes. They remain strong and frequent, but now they're accompanied by an overwhelming urge to push, an involuntary bearing down sensation as the baby's head presses against the pelvic floor and rectum. This reflex is powerful and difficult to resist. The mother typically pushes during contractions, using her abdominal muscles to increase intra-abdominal pressure and aid the uterus in expelling the baby. The baby continues its descent through the birth canal, the term used to describe the combined passage of the cervix, vagina, and vulva. The perineum, the area of skin and muscle between the vaginal opening and the anus, stretches considerably to accommodate the baby's head. In some cases, the tissue tears, resulting in a perineal laceration. In other cases, a deliberate incision called an episiotomy is made to enlarge the opening, though this practice has become less common as evidence suggests that controlled tearing often heals better than a surgical cut. As the baby's head descends, it reaches a point where it remains visible at the vaginal opening even between contractions, a moment called crowning. This is often the most painful part of labor, as the perineum stretches maximally. Shortly after crowning, the head delivers, usually face down. The birth attendant may suction the baby's nose and mouth at this point to clear away mucus and amniotic fluid. After the head is delivered, the baby's body rotates, a maneuver called restitution and external rotation, which aligns the shoulders with the widest diameter of the pelvis. With the next contraction, the shoulders deliver one at a time, the anterior shoulder first, followed by the posterior shoulder. Once the shoulders are out, the rest of the body follows quickly, often in a slippery rush. The baby is born, though still connected to the mother via the umbilical cord. The newborn's first moments outside the womb are critical. The transition from fetal to neonatal life requires rapid and dramatic physiological changes, particularly in the respiratory and cardiovascular systems. In utero, the fetus didn't breathe air. Gas exchange occurred through the placenta, and the lungs were filled with fluid. Within seconds of birth, the baby must begin breathing, filling the lungs with air and initiating gas exchange independently. The first breath is triggered by several factors: the physical stimulation of birth, the cooler temperature of the outside environment, and the sudden loss of placental support all contribute. A buildup of carbon dioxide in the blood stimulates the respiratory center in the brainstem, prompting the diaphragm to contract. The baby draws in air, often with a cry, and the lungs expand. The first few breaths require significant effort, as surface tension in the fluid-filled alveoli must be overcome. Surfactant, produced in the final weeks of gestation, reduces this surface tension, preventing the alveoli from collapsing and making subsequent breaths easier. As the lungs inflate and begin functioning, the cardiovascular system undergoes profound changes. In fetal life, blood bypassed the non-functional lungs through two shunts, the foramen ovale, an opening between the right and left atria of the heart, and the ductus arteriosus, a vessel connecting the pulmonary artery to the aorta. These shunts allowed blood to flow from the right side of the heart to the left side and into the systemic circulation without passing through the lungs. With the baby's first breaths, the lungs expand and pulmonary vascular resistance drops dramatically. Blood begins flowing through the pulmonary circulation, picking up oxygen from the alveoli. As pressure in the left atrium increases due to increased blood return from the lungs, the foramen ovale closes functionally, though anatomical closure takes longer. The ductus arteriosus constricts in response to increased oxygen levels in the blood, and over the following days and weeks, it closes permanently, becoming a fibrous ligament called the ligamentum arteriosum. These changes redirect the entire pattern of circulation. The placenta, which provided oxygen and nutrients, is no longer part of the circuit. The lungs take over gas exchange, and the heart shifts to its postnatal configuration, pumping deoxygenated blood to the lungs and oxygenated blood to the body. The umbilical cord, which connected the baby to the placenta, is typically clamped and cut within the first few minutes after birth. Though there's growing evidence that delayed cord clamping, waiting a minute or two, allows additional blood to transfer from the placenta to the baby, providing extra iron stores. Once the cord is cut, the baby is physiologically independent, no longer reliant on the mother's body for oxygen, nutrients, or waste removal. The third stage of labor, the delivery of the placenta, begins after the baby is born and ends when the placenta is expelled. This stage is usually the shortest, lasting 5 to 30 minutes. After the baby is delivered, the uterus continues to contract, though the contractions are less intense than before. These contractions cause the placenta to separate from the uterine wall, a process called placental separation. As the placenta detaches, the blood vessels that connected it to the uterus are torn and bleeding occurs. However, the uterine contractions compress these vessels, acting as a natural tourniquet and limiting blood loss. Signs that the placenta has separated include a gush of blood, lengthening of the umbilical cord as the placenta descends, and a change in the shape of the uterus from discoid to globular. The mother may be asked to push gently to help expel the placenta, or gentle traction may be applied to the umbilical cord while supporting the uterus to prevent inversion, a rare but serious complication in which the uterus turns inside out. Once delivered, the placenta is examined to ensure it's intact. Retained placental fragments can cause postpartum hemorrhage or infection, so it's important to confirm that the entire placenta and membranes have been expelled. The placenta, which served as the interface between mother and fetus for 9 months, is no longer needed and is typically discarded, though in some cultures it holds symbolic significance and may be buried or otherwise honored. After the placenta is delivered, the uterus should contract firmly, a state called a well-contracted uterus. These contractions are essential for controlling bleeding. The site where the placenta was attached is essentially a wound with open blood vessels. Uterine contractions compress these vessels, reducing blood flow and allowing clots to form. If the uterus fails to contract adequately, a condition called uterine atony, excessive bleeding can occur. Uterine massage, medications that stimulate contractions, or other interventions may be necessary to control the hemorrhage. The immediate postpartum period, the first few hours after delivery, is a time of close monitoring. Blood pressure, heart rate, and uterine tone are checked frequently. The mother is observed for signs of excessive bleeding, and the perineum is inspected and repaired if lacerations occurred. The baby is assessed using the APGAR score, a quick evaluation performed at 1 minute and 5 minutes after birth that assesses heart rate, respiratory effort, muscle tone, reflex response, and color. Each parameter is scored from 0 to 2, with a maximum score of 10. Most healthy newborns score 7 to 9 at 1 minute and 8 to 10 at 5 minutes. Lower scores may indicate a need for resuscitation or further evaluation. The hours and days following birth are a period of adjustment for both mother and baby. The mother's body begins the process of involution, the return of the uterus and other reproductive organs to their pre-pregnancy state. The uterus, which at term weighed about 1,000 grams, shrinks rapidly, returning to its non-pregnant weight of 50 to 100 grams within about 6 weeks. This shrinkage is driven by continued uterine contractions, sometimes called afterpains, which can be quite uncomfortable, especially during breastfeeding, as oxytocin released during nursing stimulates uterine contractions,